Hello. Hello, Dr. Deshpande.
Hello, sir. Hello.
Can you hear me?
Yes, sir. Yes.
Okay, thank you.
Yes, sir.
Hello.
Hello, sir. Hello.
Dr. Beg is also with me.
Yes, sir.
Okay, sir. Proceed.
Okay, sir.
Hello.
Video is stopped. Video. Hello.
सर नमस्कार सर.
हा नमस्कार नमस्कार.
गुड मॉर्निंग सर. गुड मॉर्निंग. गुड मॉर्निंग.
गुड मॉर्निंग. ठीक आहे, आवाज ऐकू येतो मला?
हो येतोय सर. येतो आवाज येतोय सर. येतोय सर.
ओके ओके. काय भानुदास पेंडकर ऐकू येतं का पेंडकर साहेब?
येतं सर. ऐकू येतं.
ओके ओके.
ऐकायला येतं.
गचांडे साहेब हाऊ आर यू?
सर फाईन सर फाईन.
ओके.
हो सर थँक्यू.
सर नमस्कार सर.
नमस्कार नमस्कार.
वर्जे सर नमस्कार सर.
ठीक आहे.
गणेश आपल्या युट्यूब वर पण टाक असेल तर.
फेसबुक
सर स्क्रीन शेअरिंग सगळ्यांनी केली आहे का?
नाही गणेश आहे सध्या.
अच्छा अच्छा. गणेश होस्ट आहे का? बरं ठीक.
सर मी तुम्हाला होस्ट बनवतो आता सर.
हा
मराठी सर.
ओके.
हो.
हो तर ते ऑप्शन पाहावं लागेल.
फेसबुक लाईव्हचं करायलो मी 2 मिनिटात करून टाकतो.
हो 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 कर.
Good morning, everybody.
गुड मॉर्निंग सर.
गुड मॉर्निंग
गुड मॉर्निंग सर. गुड
Can you
मॉर्निंग.
hear me, no? Yeah, I have just sent my presentation to the principal. No, not the principal.
Good morning
Yes.
to all.
Good morning. In case if my presentation doesn't open, you can open it from there. I send it to the HOD.
ओके ओके सर नो प्रॉब्लेम.
But I'll try opening first.
Yeah.
Good morning, Dr. Oding, sir.
Good morning, sir.
Good morning.
Good morning, sir. Good morning.
Dr. Bhale speaking.
Dr. Bale, how are you?
I am fine, sir. Oh, yeah, you are full beard. Nice look, sir. Nice
Good
look.
morning.
Good morning. Good morning.
वकील साहेब अग्रवाल नमस्कार.
Good morning, everybody.
गुड मॉर्निंग सर.
सर गुड मॉर्निंग.
गुड मॉर्निंग.
मीटिंग आहे. मीटिंग.
What?
गुड मॉर्निंग
इकडे ये.
सय्यद सर. गुड मॉर्निंग.
Good morning, Gachande sir आणि गवई sir.
मॉर्निंग सर. गुड मॉर्निंग.
हे बघ की कसं गोव्याला जाऊन बसलो.
Participants are requested to mute their
दिसायला हे बघ की
करता येते तसं करून दाखवू का मी तुला?
अरे पागल लाईव्ह चालू आहे मीटिंग सगळी.
म्यूट करा सगळ्यांनी.
न्यूयॉर्कचं कोणीतरी सर आहे. न्यूयॉर्क तिथले सर आहेत.
करा.
मराठी सत्तांना म्यूट करा. जाधव सर,
हा सर.
यू मे स्टार्ट नाउ.
ओके. प्राचार्य आहेत का सर ऑनलाईन? मराठी सर आहेत का?
आहेत.
हा सर, एक मिनिट, होस्ट व्हायचं आहे मी अजून.
बर.
ओके सर.
झाले होस्ट सर?
हो केलं, तुम्हाला अलो केलं.
ओके. गुड मॉर्निंग गवई सर. गुड मॉर्निंग. गचंडे सर चालू करायचं?
हो चालू करा.
Good morning and all welcomes to student researchers, faculty members and all participants. Myself, Dr. D.M. Jadhav, Assistant Professor, Department of Botany, Microbiology and Biotechnology, welcomes you all on this occasion of national level Dr. K. Deshpande Memorial Lecture Series 2021. In the honor of late Dr. K. Deshpande, our department organizes lecture series every year. Since last year, because of COVID-19 pandemic, we are organizing this lecture series through online virtual mode. For this lecture series, we have with us Honorable President of Nanded Education Society and inaugurator of today's program, Dr. Venkateji Kabde sir. Sir, I request you to preside over this program as President.
Okay,
Honorable
thank you.
Vice President of Nanded Education Society, CA Dr. Praveen Patil sir. Honorable Secretary of Nanded Education Society, Shamal Patki Madam. Honorable Joint Secretary, Sri Praful Kumar Agrawal sir. Principal, Nanded Education Society, Science College, Nanded, Dr. D.U. Gavai sir. We have Dr. B.F. Rodriguez sir, Senior Professor, Department of Botany, Goa University, Goa. Dr. Sanjay K. Sharma sir, Professor, JECRC University, Jaipur, Rajasthan. Dr. S.S. Varje sir, former Head of Department of Botany, Yeshwant Mahavidyalaya, Nanded, and one of the student of Dr. K. Deshpande sir. I welcome you all on this occasion. Again, I take the opportunity to welcome you all virtually.
Awaz,
Cannot
cannot hear.
Yes, I am audible, sir.
Yes, yes. You are.
Yeah. Yes. So I welcome you virtually, sir, on this occasion of national level Dr. K. Deshpande Memorial Lecture Series. First, we will give, we will offer some floral bouquet to Pujya Swami Ramanand Tirtha, founder of Nanded Education Society, by offering a garland, virtual garland, floral bouquet, and igniting Samai.
Maximum volume.
Thank you, sir. Welcome again. I welcome virtually Honorable Dr. Venkatesh Kabde, sir, President of Nanded Education Society. Honorable Vice President, Nanded Education Society, CA Dr. Praveen Patil, sir. Honorable Sau Shamal Patki, Madam, Secretary, Nanded Education Society, Nanded. Honorable Praful Agrawal, sir, Joint Secretary, Nanded Education Society, Nanded. Principal, Dr. D. Gawai, sir. Our today's guest speaker, Professor B.F. Rodriguez, sir, Senior Professor, Department of Botany, Goa University, Goa, and President of Mycological Society of India. Another guest speaker with us, Dr. Sanjay K. Sharma, Professor, JECRC University, Jaipur, Fellow Royal Society of Chemistry, UK, and Green Chemistry Series Editor, Springer, UK. With this short virtual welcome, now I request Professor Dr. B.D. Gachande, sir, Head of Department, to introduce department. Dr. B.D. Gachande, sir.
Yes. Dr. D.M. Zado, thank you. Good morning to all of you. On behalf of Department of Botany, Microbiology and Biotechnology, Science College Nanded, I welcome you to all on this occasion of national level Dr. K. Deshpande Memorial Lecture Series 2021. All of you know that whole world is under threat of pandemic, that is COVID-19 pandemic. Therefore, since last year, we are going to organizing this event on a virtual platform. Honorable Venkatesh Kabde, President, Nanded Education Society, Nanded. Honorable CA Dr. Praveen Patil, sir, Vice President, Nanded Education Society, Nanded. Honorable Shamal Patki, Madam, Secretary, NES Science College, NES, Nanded Education Society, Nanded. Honorable Sri Prafulla Kumar Agrawal, Joint Secretary, Nanded Education Society, Nanded. Honorable Dr. D. U. Gawai, Principal, Science College, Nanded. The chairman of this function, Dr. Venkatesh Kabde, a well-known cardiologist, ex-member of Parliament and President of Nanded Education Society, Nanded. Honorable Vice President, Honorable Secretary, Honorable Joint Secretary, Nanded Education Society, Nanded. One of the senior most professor, a well-known prestigious society, Mycological Society of India, President Dr. Rodriguez, sir. Another chief guest and today's speaker, Dr. Sanjay Kumar Sharma. Honorable Dr. S.S. Wadje, sir. All the dignitaries, delegates, head of the departments, faculties, participants, and students from all over the country. The organizing secretary, Dr. P.S. Borkar, Dr. Praveen Paul, Dr. R.V. Sangvikar, executive members of organizing committee, all research scholars and PG students of our department and other departments of our college and other colleges. I heartily welcome to all of this Dr. K. Deshpande Memorial Virtual Lecture Series. I am here to introduce the department. Now this department is being organizing this lecture series since 2008. This department is one of the foremost and leading department in our college. The department is started with the inception of college with PG in botany since 1970 by late Dr. K. Deshpande, sir. The department has indelible impression in the field of teaching and research in plant sciences. The department is recognized as a research center since 1970. The tradition of merit is maintained by the faculty and students. There are 64 students have been awarded PhD degree from this department, and PG students continuously maintain the position in the university merit every year. more than 360 research papers have been published so far. The department now has 9 full-time faculty members and 10 core teachers with different area of specialization. Their contributions are reflected in the form of research. They publish their research papers in research journals of national and international repute, books, chapters in books, huge number of major and minor research projects funded by University Grants Commission, DST, CSIR, etc. are completed by the faculty to pursue excellence in the field of plant science research. The department has adequate infrastructure with spacious laboratories, research laboratory cum plant disease clinic. The department is rich in well-equipped laboratories with ICT-enabled classrooms. Department has well-established botanical garden, medicinal plant garden, and a greenhouse for maintenance of some rare and endangered plants. Museum is enriched with many rare specimens. Department runs 4 PG courses, that is botany since 1970, one of the grant in course, that is MSc botany, then biotechnology since 2010, microbiology since 2019, and another postgraduate PG course in herbal medicine funded by UGC, University Grants Commission, New Delhi. Now I wish to tell some few words regarding K. Deshpande sir. The full name of Dr. K. Deshpande sir is Dr. Kalidas Shankarrao Deshpande, one of the mycologists and plant pathologists from Marathwada region of Maharashtra. He born at Devni, Latur, to a landlord family of Palam, Parvani. He completed his schooling education in Palam and Gangakhed, district Parvani. He established a PG course in botany in this college in 1970 and later became principal in 1994. He awarded with RISC scholarship of Hyderabad State. He completed his graduation with botany, scoring position in Marathwada University, Aurangabad, and master degree in botany from the same university, securing first position. He completed his PhD degree under the guidance of Dr. K.B. Deshpande in 1967 from the same university. He joined the Science College Nanded as founder, head of the Department of Botany, and started PG in Botany in 1970. 19 students have been awarded under his guidance for PhD degree. He did his postdoctoral degree under the supervision of Professor RKS Wood, doctoral advisor to many leading mycologists and plant pathologists at Imperial College of London. His work carries basic mycology and plant pathology, physiology of microbial infection, cell wall degrading enzymes of plant pathogens, plant disease control, and discovery of some novel unknown plant diseases and pathogens. He initiated an exhaustive study of fungal pathogens of banana and diseases caused by them in this region. His contribution in establishment of modern biology at University Subcenter Nanded University, later became School of Life Sciences at SRTMUN Nanded. He was honored with an eminent scientist on the occasion of Diamond Jubilee celebration of Saraswati Bhuvan Education Society, Aurangabad. He was honored with Nanded Bhushan by Nanded Municipal Corporation Nanded for his contribution in the field of education and social commitment. He was also president of Marathwada Botanical Society, Aurangabad. He was awarded with 3 gold medals and appreciation in 8th World Congress on Ecology and Environment, New Delhi. Giving to his contribution in the field of education, research and extension, he was nominated by Government of Maharashtra as a member on the Planning and Development Board, Shivaji University, Kolhapur, Maharashtra. He was married with Dr. Usha Deshpande. She was a reader in botany in Science College Nanded. She has assisted him in teaching several courses and she was eventually a member of the departmental staff. They have a son, Dr. Rajesh Deshpande, working as a well-known neurologist at Ruby Hall Clinic, Pune, and daughter Tanuja Patki, an engineer living in Bangalore. As well, proud I felt proud as one of the departmental colleague of Dr. Kesh Deshpande for 8 years. He was always encouraging the students when things were not good. He was source of inspiration and creation of and ideas. He was a master in subject, best teacher, motivator, and a kind person. His students framed a felicitation committee at the time of his superannuation. Dr. S.S. Wadje, as a chairman of this committee, felicitation committee, Dr. D.S. Mukadam, Dr. M.M. Beg, Dr. R.B. Katti, Dr. Joseph, R.D. Joseph, Dr. Ila Papu, Dr. Anuradha Sinde, Dr. T. Prasanna, and Principal Dr. Gavi. The felicitation committee sponsored a gold medal to a university topper in MSc Botany in every year. In his lifetime, he wanted to establish and develop biotechnology park in this college, but unfortunately, after submission of this proposal to the funding agency, immediately he passed away on 16th October 2000. With these few words, I salute to late Dr. Kesh Deshpande sir to his contribution in the field of mycology and plant pathology. Thank you very much for your participation in this event and patient listening. I again welcome to all the participants, delegates, dignitaries, all chief guests, Professor Rodrick sir, Professor Sanjay Kumar Sharma sir from Jaipur for accepting our invitation and given consent to deliver a guest lecture on this occasion. Thank you all. Thank you.
Thank you, Professor Deshpande sir, for introducing department and briefing about Dr. Kesh Deshpande sir. Now, I request one of the student of Dr. Kesh Deshpande sir, Dr. S.S. Wadje sir, to say something about Kesh Deshpande sir on this occasion. Wadje sir.
Good morning, all of you.
Good morning, sir.
Being a first student of the department and first student of the Dr. K. Deshpande sir, also alumni of the department, I welcome you all to the program. Honorable Dr. Kabde sir, Honorable Praveen Patil ji, Madam Shamal Patki, Agrawal ji, eminent speaker Bernard Rodriguez, Dr. Sanjay Sharma ji, departmental staff, Dr. Gachande, Principal Dr. Gavai, Dr. Gachande, Head Department of Botany, Dr. Marathi, Dr. Jadhav, Dr. Sangvikar, Dr. Vinayak Chavan, and rest of the staff. I am here as to welcome you and to say something about Dr. K. Deshpande being a first student of Dr. K. Deshpande. Actually, Dr. K. Deshpande is founder, he was head of the department, PG Department of Botany Science College, Nanded, and also become principal of that college later on. I pay my sincere tribute to Dr. K. Deshpande. He was very famous teacher. He was plant pathologist, mycologist, great scientist of international repute. He has produced number of PhD students, over 20 PhD students, and published nearly more than 100 research papers. He inspired number of students from the department for great work. Later on, Dr. Deshpande established several laboratories fully equipped laboratories in the college, research laboratories. He actually completed several projects, research project funded by the UGC, DST, CSR, etc. He also worked on number of planning bodies of the government of Maharashtra. He brought name and fame to the department, and the great foundation he laid to the department. As department is standing right now, and department is working in a very good condition now. I wish best to the department, and I wish best to the today's program. Thank you all.
Thank you, Dr. Varje sir. Now I request Principal Dr. D.U. Gavai sir to introduce college.
President of today's inaugural function, Honorable Dr. Venkatesh Kapde sir, who is President of Nanded Education Society, Nanded, was an ex-MP and well-known cardiologist of this region. Vice President of Nanded Education Society, CA Dr. Praveen Patil sir, Secretary of Nanded Education Society, respected Shamal Patki Madam, Joint Secretary of Nanded Education Society, Prafulla Kumar Agrawal sir, resource person, Dr. Bernard Rodriguez sir, Senior Professor, Department of Botany, Goa University, Goa. Another resource person, Dr. Sanjay K. Sharma sir, Professor, JE CRC University, Jaipur. Distinguished guests from all over India, organizers of this Dr. K. Deshpande Memorial Lecture Series, convener Dr. B.D. Gachande, Professor and Head, Department of Botany, Microbiology and Biotechnology of this college, Organizing Secretary Dr. P.S. Borkar, Associate Professor in Microbiology, or another Organizing Secretary Dr. Praveen Paul, Assistant Professor in Microbiology, Dr. R.V. Sangvikar, Assistant Professor in Botany, all the committee members, all the colleagues, all my colleagues from Botany Department and Chemistry Department, Dr. A.T. Shinde, Dr. D.M. Zado, Dr. V.B. Chavan, all the students of PG Botany from our institute, and the students of Dr. K. Deshpande sir. On this occasion, it gives me a great pleasure to extend the heartiest welcome to Senior Professor Dr. Bernard Rodriguez sir. A hearty welcome to distinguished speaker Professor Sanjay Sharma sir. Here, I will introduce our college in brief. Our college is a single faculty college run by Nanded Education Society, Nanded. The Nanded Education Society, Nanded is one of the premier institute in this region, established by Pujya Swami Ramanand Tirth, who was a great visionary and leader of Hyderabad liberation struggle. Our college is reaccredited with A grade by NAC in third cycle with CGPA 3.38. It is recognized by the DST for fifth program. We received college with potential for excellence status by UGC New Delhi. NIRF ranking of the college is 72. There are 15 subjects offered at UG level and 11 subjects at PG level. Out of these 11 subjects, there are 2 PG subjects, MSc Botany and MSc Mathematics. They are on granted basis. Research activity, research ambience is very well established in the college. Our faculty, they are engaging themselves in the research, and therefore 4 major projects and 18 minor projects, they are completed in last 5 years. There are 23 research guides at present, and more than 40 students they awarded PhD in last 5 years. 40 students qualified NET, SET, GATE, JRF examination, and we have the tradition of university ranker. In last 5 years, 40 students they received a university rank in last 5 years. Our college is always at the forefront in organizing such type of seminars, conferences, workshop, extension lectures to enhance the teaching and learning competency of learners. This lecture series has been instituted for a worthy purpose. The key to the success of any organization entails dissemination of knowledge. This requires interaction with academics, industry, government, students, and various other institutions. The Department of Botany has been conducting memorial lecture series since 2008 in the honor of Dr. K. Deshpande sir, who made great contribution to teaching and research in botany. Dr. K. Deshpande was a teacher for excellence. He was not confined to academics only. He was also a good administrator, worked as a successful principal of this college. He was a man of honesty and integrity. He was a devotee worker in the field of mycology, plant pathology, particularly in the physiological plant pathology. Enzymes, toxins is the area for his research, and soil microbiology. We have seen his greatness, determination, and commitment to the farmers. Because of his outstanding work in the field of research and society, he has been awarded Nanded Bhushan Award by Nanded Wagala Municipal Corporation, Nanded. I pay tribute to my Guru for his inspiration, motivation, and support to build my career. I wish a grand success for this lecture series. Thank you, one and all. Thank you very much.
Thank you, Principal Gawai sir. Now I request President of today's function, Honorable Dr. Venkatiji Kabde sir, to declare inauguration of this lecture series and deliver presidential address. Dr. Kabde sir.
Thank you. Honorable office bearers of the Orchid Education Society and the guest of honors and the national level speakers, Dr. Bernard Rodriguez, Professor of Goa University and President of Mycorrhizal Society of India, Dr. Sanjay Sharma, Professor of Jabal University, and those people who have participating in this national level conference. I also appreciate Dr. Deshpande Memorial Lecture organizers and the organizing committee, Dr. Sangvikar, Dr. Paul, Dr. Borikar, and convener, Dr. Deshpande, and all the participants who are participating from all over the country. Particularly, I would like to appreciate Dr. Gawai, who has taken a very great role in organizing this conference. I think our society has lost a great personality in Dr. K.S. Deshpande's death, because he was a great scientist. He was a scientist par excellence, a very great teacher and a great principal. And I think it is because of the heart attack that he died suddenly and in a premature fashion, and the contribution to our society has been greatly lost because of his death. But he has left behind a legacy of knowledge and students who are conducting research as well as educational activities in our institution in a great way. I pay my tributes to Dr. K.S. Deshpande and his work, and I wish all the members of his family, many members are still alive in the form of Dr. Rajiv Deshpande and others who are doing a great job in medical and other fields. Dr. Deshpande has produced such wonderful students and research workers that whatever amount of details I give here would be inadequate. And I think this is a great tribute to his memory, that we are organizing this activity every year, and it is the pursuit of knowledge, which is the aim of our society. The aim of our society is to inspire young people for research, and also at the same time to take care of the people who are lagging behind. This work has been going on for the last so many years and will continue forever. And I think the basic research work, which was the great slogan of Dr. Deshpande in his life, that will be carried on by our institution. I think this is a great national seminar. I am not a very competent authority to say anything on this subject. Is a mycorrhizal fungi, which is mycorrhizal fungi, and we have a great authority in the form of Dr. Bernard Rodriguez, who is going to speak on this topic. It is a subject of great interest. As a doctor, I know that the fungus has created a lot of problems. Mucormycosis, you have been reading about it day in and day out. Fungi have a great role, and some of the plant roots that they have been studied for their organisms, fungi, and how they react with the soil, and what is the absorption of nitrates and the water, and all this will sort of impart great knowledge about our soil conservation, and how it's a national aspect also to this subject. So I think Dr. Rodriguez, his contribution in this matter will be of great help to the students and research workers. And the other subject is also of great importance: research publications, ethics, and misconducts. It's of very vital importance. And when we are talking about the research, the integrity in the research is very important. It has got two main aspects. One is the research integrity violation that is included in this subject, which is of paramount importance, because whatever research you do, if it is not done with the integrity, I think there is a lot of damage done to the research. The other thing is about the publication ethics, which is also important, but is of secondary importance, because somebody just copies some research subject and then gives his name, or gives the name of his teachers who are supervisors, but still he gives the name. That is a sort of minor error in the publication ethics. So these are the very two vital aspects of research in the research publications, which have to be taken care of. And I think we have authorities, great authorities in the form of Dr. Bernard Rodriguez and Dr. Sanjay Sharma, who are going to enlighten all the participants from all over the country on these two very, very important subjects. So I welcome them and all the participants who are participating in this webinar, and wish them well. Thank you very much.
Thank you, Dr. Kabde sir. Now, so we'll move towards the next part of our lecture series. I request Dr. P.S. Borkar madam to introduce today's guest speaker, Professor B.F. Rodriguez sir. Borkar madam. Madam, unmute.
मॅडम आहेत ना, अनम्यूटच आहेत मॅडम. बोरकर मॅडम, हॅलो. सर आवाज येत आहे?
हा, आवाज येते.
बोरकर मॅडम अनम्यूट आहे, जॉईन आहेत मॅडम. एक मिनिट कॉल करतो.
Something technical issue might be there with Dr. Borkar madam. Yeah, just a minute.
Hello, sir.
Just a minute.
Hello, sir.
हॅलो. हा हा सर, मॅडम बोलतायत. हा मॅडम चालू करा.
गुड मॉर्निंग टू ऑल.
ऐकू येतंय का?
Yes.
Okay.
येतोय सर आवाज?
हो येतोय.
Yes, ma'am.
Okay.
It gives me immense pleasure to introduce one of the most eminent personalities of today's national level, Dr. K.S. Deshpande Memorial Virtual Lecture Series. I am Dr. Preeta Borkar, Associate Professor in Agricultural Microbiology, Department of Botany, Microbiology and Biotechnology, Science College, Nanded. I am here to introduce Professor Dr. Bernard Rodriguez, Senior Professor, Department of Botany, Goa University, Goa. Presently, is Directorate of Internship, Incubation and Industry Partnership from 2018 and President, Institutions Innovative Council, Goa University since 2018 till date. About his honors and awards, he is twice recipient of Young Scientist Research Project Award from Department of Science and Technology, New Delhi. He has received Excellence Award from Tanzanian Parents Network Foundation in 2010, Professor Bhide Memorial Award by Indian Society of Mycology and Plant Pathology in 2016. He has received Best Teacher State Award from Government of Goa in 2017 and Dr. B. Agnihotridu Memorial Award by Mycological Society of India in 2019. He is a Fellow of Mycological Society of India, Fellow of Indian Society of Mycology and Plant Pathology, and under his stewardship as President of Institutions Innovative Council. Goa University was awarded 4 stars in 2018-19 and 4.5 stars in 1920 by MHRD, New Delhi. And at present, he is President, Mycological Society of India. About his academic achievements, he has completed a dozen of major and minor research projects. He has attended over 165 national and international conferences, workshops, and seminars. He has delivered plenary and lead lectures. Besides, he has chaired several scientific sessions. He has published unqualified research papers in national and international journals of repute, edited more than 5 books, and so far guided 12 PhDs, and presently he is guiding 5 students. He is on editorial board of many journals, life member of Botanical Society of Goa, Mycological Society of India, Indian Society of Mycology and Plant Pathology, Indian Mycological Society, and Indian Society of Plant Pathologists. He is a reviewer for number of national and international journals. Sir, his biodata is so huge that what I have presented is just a water drop in the ocean. Thanks for giving me this opportunity to introduce the renowned mycologist and president of Mycological Society of India, a prestigious society of academicians, that is mycologists from all over the world. Thank you, sir.
Thank you, Dr. Borke madam. Now I request Professor B.F. Rodriguez sir to start the lecture.
I'll just share this presentation.
Yes, sir, yes.
Some problem, I think. I'll just.
Yes.
Sir, I have allowed you to share the screen. Sir, please share screen.
That's what I'm trying to do.
Okay.
I think I'll have to sign in again. Looks like.
What problem, sir?
That I'm not getting your screen, the meeting screen.
Okay. Can I share your screen, sir? Your presentation from here?
Yeah, but I'm not getting your screen, the main screen.
Sir, you just
Shall
click
I log in again?
Hi, sir. You are.
Yeah, no problem, sir. You just log in again.
I'll do that.
No, sir. You just at right hand corner, the view is there, gallery view and speakers view. You just allow a gallery view so that you can find out whatever.
Now I got this one. Then share screen.
Sir, at the bottom, the option is there, no?
Share screen.
You are visible to us. Yes, you are visible to us, no? Just see at the bottom.
Share screen. Share. Is it
no,
shared?
Sir, no, not yet.
No. Let you go.
Green color.
So at the bottom, screen share option is there on your, because you are visible to us. You are online already. Yes.
Yes. Sir, please double click so that it enter into full screen.
Slideshow mode.
Okay.
Again, scene screen has stopped.
Let's just stop here. I'll go to my presentation.
You do it for one more time, sir.
Yeah, yes.
Just your screen very visible to us.
Yeah. Presentation is visible, sir.
No, no, sir. No,
No, no, sir.
Presentation.
No, no. Just you are live, sir. You are visible to us. So just you open your presentation and just screen share it. At the bottom, a green button is there, screen share. You just click on it. Automatically your laptop or it will get share your screen.
No, I want to get your site. It came and it disappeared again.
Okay. So,
The present meeting site.
okay.
Sir, you just tap on the screen so that you screen share.
No, screen share. Yeah, I think now you'll get it.
Okay.
Is it clear? Is it there?
Now it is sharing right now.
Is it there, sir?
Now you are sharing. Yes, sir. Yes, yes, sir.
Yes, sir.
Can you see this?
Yeah. We can see.
Sir, please make it slide share.
Full screen. Full screen.
Sir, please press F5 button on your laptop or computers.
You can see the screen.
Yeah. Screen share is there, but it is not in a full form, sir. You just press F5 button.
Again, it has got disconnected.
So इथून करू का मी share?
बराबर.
Okay. Sir, can I share your presentation from here, sir? Rodriguez, sir.
I think sir left झाले वाटले. पुन्हा त्या region होऊ द्या.
Okay.
Hello.
Yes, sir. You are audible to us.
Sorry, whether you can open the presentation would be better.
Okay.
Okay.
Some problem here, I think. Yeah, now
Yeah.
it is open.
मराठी sir, तुम्ही open करा.
I'll try once again. That will be better. Yes.
Yes.
Okay. Yeah, I'm sorry for the inconvenience that is caused. Well, Honorable President, Dr. Venkatesh Kagde sir, and all the other honorable members of the Nanded Education Society, Nanded, Principal Dr. Gavai sir, Head of the Department, Dr. Gachande sir, and all the faculty members of the departments of botany, microbiology, and biotechnology, and other departments. And I thank you all for giving me this privilege to deliver this late Dr. K.S. Deshpande Memorial Lecture Series. Well, I take this privilege to pay my humble tributes to late Professor K.S. Deshpande, who was a towering personality and has great contribution in the area of mycology and plant pathology. The topic, before that, I would also like to thank Borkar Madam for nicely introducing me to the August gathering. The topic allotted to me is mycorrhizal fungi. Talking on mycorrhizal fungi, I'll be talking general aspects of mycorrhizal fungi, their classification, characteristic features, taxonomy and identification, inoculum production, and some examples of inoculation studies conducted by me and my students. Well, to begin with this particular topic, mycorrhizal fungi. Can I have the next slide? So please. Yeah, okay. Hello. Am I audible?
Yes, sir.
Yes, sir. Yes. I think range problem is there, sir.
Network issue is my network issue is
Can you? Hello.
Hi, yes.
सर मराठी सर आहेत का सर?
हा, लेफ्ट झाले होते, पुन्हा जॉईन केलं मी सरांना. नाही, 2 इथून जॉईन झालेत सर.
असं आहे का? अच्छा अच्छा. एक
गचंडे सर, एकदा फोन करून पहा ना सरांना. सर लेफ्ट झाले काहीतरी, बहुतेक नेटवर्क इश्यू आहे सरांकडे.
सर जॉईन झाले दिसाले.
Now, okay. Am I audible, sir?
Yes, sir.
Okay, fine. I'll continue then. Yeah. Now coming to the historical of mycorrhizal, it was in the year 1881 that he discovered the symbiotic association in fungus and roots. A German mycologist coined the term mycorrhiza for the can I have the next slide? Hello. Am I audible? I think I'll have to switch on to another PC. Hello.
Hi, yes, sir. You are audible.
It was
Sir, please stop your video so that bandwidth will be better then.
So the mycorrhizal put on this one. Yeah. Hello.
Hi, yes, sir.
Yeah. So I will continue and then I will go move on to the another PC because there's some problem I think with this PC. I don't know why it is going on like that. Anyway, I will continue. So the mycorrhizal system consists of the plants, soil, and the fungi. So the plants are important because they are colonized by the mycorrhizal fungi, and soil is equally important because all this happens in the soil. Of course, there have been experiments done where the mycorrhizal fungi can be cultured without soil. So I will come to that a little later. So the next slide, please. Yes. So now the symbionts contribute to the nutrition of the partner. There is a bidirectional flux. That is, both the partners are benefited. The fungus is benefited as well as the plants are benefited. Now the plants are benefited because these mycorrhizal fungi, they absorb nutrients from the soil, especially phosphorus, nitrogen, and others, and make it available to the plant root. So the plant is benefited, and in turn, the fungus gets carbohydrates or the carbon compounds from the plant. Since the plant is unable to produce this, since it is there's no photosynthesis in the fungi. So that's how there is a bidirectional flux which is leading to symbiosis. So the next slide, please. Yeah. Now some of the facts of mycorrhizal fungi. They constitute a special group of among root inhibiting fungi. They are universal in occurrence. The fossil records of mycorrhizal fungi indicate that these fungi are more than 460 million years old. Now that was a time 460 million years back. There was a transition from aquatic to transition of plants from aquatic to terrestrial habitat, and it was during this time that mycorrhiza has played a great role in the survival of these plants on the land. That is because just you can imagine the conditions, the temperature that was existing on the planet where there were no plants at all. So that is the time the mycorrhizal fungi have assisted the plants so that the vegetation came up, and after vegetation, the animals and human beings. If today we find that there are human beings and the evolution of other organisms we should, these mycorrhizal fungi are very important. Now there have been studies done on that whenever the mycorrhizal fungi were absent, then there was failure of afforestation programs. Many of the afforestation programs, especially with the exotics which were tried, it was found that these exotic species failed to survive in the new habitats, and therefore these the reason was that there was no mycorrhizal inoculum. It was only after the mycorrhizal inoculum was transferred and used for these saplings of exotic species, then they survived, indicating that mycorrhizal fungi play a very important in their role. And without their absence, these exotic species would not survive in new areas. So that is the importance of mycorrhizal fungi. Can you move to the next slide, sir, please? Yeah. Now coming to the classification of mycorrhizal fungi, it is based on the infection anatomy. We have two main categories, namely that. Now we take up the ectomycorrhizae first, and I have done few studies on ectomycorrhizal fungi that also I will show. So they are common among the forest and ornamental tree species like the members of Pinaceae, Salicaceae, Betuliaceae, Fagaceae, Tiliaceae, Rosaceae, and so on. Which is the important characteristic of the ectomycorrhizal fungi. Second.
Hello, sir. Rodri, sir, you are not audible, sir.
ते म्यूट दिसले सर, म्यूट झाले टोटल. सर दोन हे जॉईन झालेत का?
नाही, एकच आहे. आले.
दोन वेळा म्यूट झाले. Okay, now it's clear, I think.
Sir, Rodri, sir, please.
The roots of. Shall I go to another PC?
No, sir. Please stop your video so that your voice is clear. Then, I think internet problem, range problem is there. Okay.
It goes off suddenly. That's the problem. Now I'm clear.
Yes.
Okay. Okay. Yes, sir.
Right.
Okay. So if you see the ectomycorrhizal roots, they are short, they are swollen, dichotomously branched, and they have varied colorations. You can see here that these colors of the roots, they vary. Now the color of the roots is because of the type of my ectomycorrhizal fungus. So based on the color of the roots, you can identify the fungus. That is the peculiarity of ectomycorrhizal fungi. The reason is the roots are covered by the hyphal mantle, and the hyphae, the mantle that gives color to the roots, have white and black and orange and different type of colors. Now coming to the isolation and pure culture of ectomycorrhizal fungi. Isolation is you isolate by using the routine microbiological techniques where the roots of these plants, which are ectomycorrhizal, they are taken and they are cultured in a petri plate, and then the fungus would grow and multiply, and that's how the pure culture is prepared. But the problem is that there are quite many species which are not able to grow in culture. So the inoculum production is simple for those species where it is possible to multiply or to grow in culture. Can I have the next slide, sir, please? Yeah, you can see that this is the mantle or the hyphal sheath. It's called as the fungal sheath, which is formed in peculiar feature of the ectomycorrhizal fungi. See, you can see the sheath here covering the root, and then inside you find the presence of a Hartig net. This is the Hartig net. It's very clear here. It's in the form of a net that was discovered by Hartig. Therefore, it's called as the Hartig net. So the presence of mantle or the hyphal sheath and the Hartig net are the characteristic features of ectomycorrhizal fungi. Can I have the next slide, sir? So that was ectomycorrhizal. Now we come to the second category of mycorrhizal fungi. This is the endomycorrhizal fungi. In that we have different types, the Orbiscular mycorrhizal fungi, which are ubiquitous in distribution. I'll be talking mainly on the ectomycorrhizal, the Orbiscular mycorrhizal fungi in this presentation. So there are very few families which may not form mycorrhizal association, this Orbiscular mycorrhizal association, such as the Brassicaceae, Chenopodiaceae, Polygonaceae, Cyperaceae. But even in these families, there are few members which produce the Orbiscular mycorrhizal association. And these fungi are placed in a new phylum, Glomeromycota. So a new phylum has been erected for these fungi, and they show intercellular as well as intracellular colonization. Now, in case of ectomycorrhizae, what we saw, the presence of Hartig net, there was only intercellular colonization. There was no intracellular colonization. Here, there is both intercellular and intracellular colonization. Isolation, the pure culture and the inoculum production is, it is soil-based. We have to depend on the spores, isolate the spores, then prepare the pure culture and the inoculum production, which is a little tedious. But once you have it, then you can always maintain the cultures. I'll discuss that in detail. Can I have the next slide, sir? The other categories includes the Arbitoid mycorrhizae, that is called as Ectendo Mycorrhizae. Now this is called as Ectendo Mycorrhizae because it is a transitional stage between Ectomycorrhizae and Endomycorrhizae. It has got these roots have both ectomycorrhizal and endomycorrhizal colonization. So there is root dimorphism. Examples like Amanita, Cotinereus, Boletus are some examples. In case of Ericoid mycorrhizae, you can see the fungal ramification in each cell. So the 80% of the cell volume is occupied by these fungal hyphae. So Pizziza is one of the examples. And then finally, the orchid mycorrhizae like Epipegon and Vanilla, they are very important, commercially important, economically important. And these orchids, they depend on mycorrhiza for their growth. Now, the entry of the hyphae or colonization is a must for further growth of these plants. The fungi that cause orchid mycorrhizae includes foams, then corticum and rhizoctonium. Next slide. You can see in the next slide, sir, that there is a. Can I have the next slide? Yes. These are the orchid mycorrhizae. Ericoid mycorrhizae. You find that there is a fungal ramification. About 80% of the volume, you can see here, it's occupied by these fungal knots. This is a section showing the amount of fungal knots occupying the cells. And you have the orchid mycorrhizae, where they produce these type of structures which are called as the pelletons. So the pelletons is a characteristic feature of the orchid mycorrhizae which are found in orchids. So when you take a root, when you stain it by using trypan blue, when you see this type of association, then you can easily say that it is the orchid colonization orchid by orchid mycorrhizae. Next slide, sir. Now coming to the benefits of Orbiscular mycorrhizal fungi, they have numerous benefits. Like first important benefit is that they help in the uptake of nutrients, especially phosphorus, nitrogen, potassium, and other micro and macronutrients, and therefore they increase the yield. So there is increased water uptake, maximum nutrient uptake. When there's maximum nutrient uptake, then it helps the plant. So in short, it helps the health of the plant. So when the plant is healthy, then it can overcome all the other stresses. Then reduce transpiration stress. Then fertilizer requirements, we use expensive fertilizers, inorganic fertilizers, so that when you use the mycorrhizal inoculum, then that is reduced. The use of expensive fertilizers is reduced. Then these fungi, they play an important role in the heavy metal toxicity. They reduce the heavy metal toxicity in the soil. So therefore, many of these mycorrhizal fungi, they are used along with the plant species which can tolerate these heavy metal sites, which when they are colonized by the mycorrhizal fungi, they add to the uptake of these heavy metals into the plant system. Not only into the plant system, but also into the mycorrhizal fungi. There are structures which are called as the orbicles and the hyphae which store these heavy metals. Then it helps to maintain the soil fertility, that is sustainability of the soil, and of course disease resistance, because when the plant is fit, the fitness of the plant is good because of mycorrhizal association, then it can overcome the diseases. Next slide. So you can see a simple experiment that we have done, where mycorrhizal fungi, when they added, then it increases the growth drastically. So you can see the increase. This is in case of chili. The growth has increased because of mycorrhization. Whereas where in these pots no fertilizers were added or no manures were added, except there was mycorrhizal colonization. Next. Oh yeah, here you can see that these are the roots, and these are plant roots again, but they are colonized by mycorrhizal fungi. So that is rhizosphere versus mycorrhizosphere. When you talk of rhizosphere, there are only the roots along with the plant, and here the roots are colonized by mycorrhizal fungi. So these mycorrhizal fungi, they have the ability, this hyphae, they have the ability to get into the soil where the roots cannot reach. So they go beyond the root depletion zone. Now, when we talk about the phosphorus is the mobility of the phosphorus is very poor. So the movement of phosphorus from the surrounding to the root zone, it takes much time. So by then, whatever phosphorus is available at the root zone is absorbed by the roots. And therefore, this creates a zone of deficiency or zone of depletion. And this depletion zone affects the plant growth. Now, this can be overcome by mycorrhizal or mycorrhization. When you have mycorrhizal colonization, these roots can go meters into the soil and absorb phosphorus that is available in the soil and make it available to the plant, which the roots cannot absorb. Next. Here also you can see in the next slide the size of the roots, the plant roots versus the fungal hyphae. The fungal hyphae are so small that they can enter into any fine root into the crevices, soil crevices, and absorb the available phosphorus. So that there could be phosphorus available in pockets where the roots cannot penetrate, and where these fungal hyphae, they penetrate and they make the phosphorus available to the plant. Next one. Now, how to identify these fungi? Now, within the roots and in the soil, how we know that the plant is mycorrhizal? So there are ways and means that these mycorrhizal fungi, they produce structures, specialized structures, like they produce these hyphae. Here you can see the linear, hyphae which are not septate, non-septate hyphae. So the mycorrhizal hyphae are non-septate. The hyphae of other fungal species like the endophytes and all, they are septate. That's why you can easily say that the colonization is mycorrhizal. Further confirmation is done by the presence of these orbicles. So these are hostoria-like structures which are found in the cortical cells, and they help in the transfer of nutrients from the plant to the fungus and from the fungus to the plant. Then again, there are other specialized structures which are called as vesicles. These vesicles may be intercalary or they may be terminal. Now here these are terminal vesicles. The function of this vesicle is that they have two functions. They store lipids and they also form into chlamydospores. Now this reproductive structures, the chlamydospores, are important because suddenly under stress condition, when there is water stress, when there is some other stress, immediately these structures would be formed and they would remain dormant in the soil. With the return of the favorable conditions, these chlamydospores would germinate. So the vesicles have double role. One role is they act as storage organ, they store lipids, and the second function is that they help in reproduction. Besides, they produce these type of structures which are called as the auxiliary cells. Now both these are auxiliary cells. These are auxiliary cells which are produced in the roots. When you stain the roots, you can see this type of auxiliary cells. They are, they are not frequently found, very rarely found. And these are auxiliary cells which are found in the soil, which have this type of brownish coloration. Now, coming to based on these auxiliary cells, you can identify the species because they may have this type of papillae. They may be, they may be having this type of appendages, which may be acuminate or finely papillate. So based on that, one can identify the species. Now, those species that produce vesicles will not produce auxiliary cells. And those species which produce auxiliary cells, they don't produce vesicles. Now, EM fungi or the Orbiscular mycorrhizal fungi, they are known as EM fungi. Earlier, they were called as VEM or VEM. V was used because of the vesicles. It was found that all these fungal species, they do not produce vesicles. And therefore, the alphabet V was deleted. So they became EM fungi. Like the members of the genera Gygaspora and Scutorospora, they do not produce the vesicles. They produce only these auxiliary cells. So based on these auxiliary cells, you can identify whether it is a Gygaspora, which species, or Scutorospora, which species. In addition to that, the mycorrhizal fungi, they produce these spores. The spores are produced in the roots as well as in the soil. So you get nicely, different spores with different ornamentation, colors, size, and so on. Next slide, please. Now coming to these auxiliary cells, we did some very interesting study on the auxiliary cells. We wanted to know what is the function of the auxiliary cells, whether they store lipids or whether they help in reproduction. So we have observed that when these auxiliary cells, these auxiliary cells we could develop in vitro culture, in the petri plates. Next slide, sir. You can see that when we tried to stain this with Sudan black, then we found this type of staining indicating that the auxiliary cells, they store lipids. So whatever the function which the vesicles they do, the same function of storing lipids is done by the auxiliary cells. So this is in case of the genera Scutorospora and Gygaspora. You can see these auxiliary cells, they are nicely growing in the culture medium in the petri plates. And whereas the other function of reproduction, we could not, so far we have not been able to identify whether these auxiliary cells, they perform the function of reproduction. And as of today, what we believe that they don't perform the function of reproduction, since the spores are produced by the members of Gygaspora and Scutorospora families. Next slide, please. Now coming to the isolation, how do we isolate? So the mycorrhizal technology is based on the fungal spores. So we try to isolate the spores from the rhizosphere. Like this is a pot which I have shown that where there is a soil, and from the rhizosphere there will be spores. And this is done by using the wet sieving and decanting technique. So wet sieving and decanting technique, and then the other methods, modification, where you use the sucrose density gradient centrifugation, and then we collect the sievings from the different sieves, and they are passed through the funnel, and these sievings are collected in the petri plate, and then the petri plates are observed for the presence of spores, and these spores are isolated by using, next slide please, by using the stereo microscope. Now this is simple. This is where the roots, we collect the rhizosphere soil, and then by using these sieves, we pass through the sieve through the funnel, and the supernatant gets collected here, whereas all the spores and debris is collected and observed under the microscope, stereo microscope. We isolate the spores. Next one. Yeah, next one. Now once you get the spores, many times people, no, the previous one. The many times what people working on mycorrhiza or the mycorrhizal researchers, what they do is, can go to the next step, please. Is what they do, yeah, yes, no, the previous one is that they take the spores which are isolated from the rhizosphere and they try to identify it, which is not the right way of identifying the spores, because the soil may contain spores which are there for thousands of years. Now what we need to get is, we need to get fresh spores, we need to get spores which are viable, and therefore we need to prepare trap cultures. So the trapping can be prepared by various means. Like you can use the rhizosphere soil where there are spores that could be used for trapping, or you isolate the spores, PM spores, and then go ahead with the trapping. Or sievings can be taken and could be used after passing through the sieves. Or you can take the entire plant along with the rhizosphere, the intact plant without disturbing the root system, place it in the pot, and then that could be useful for multiplication of the trapping the spores. Or you can use the roots of the host plant, and one can use for trapping the cultures. Now, for trapping to prepare trap cultures, the selection of the host plant is very important. Now, these are some of the features of the host plant. The host plant, first of all, it should grow fast, should show fast growth. It should adapt to the prevailing growing conditions. It should get readily colonized. The roots have to be softer. It should produce a large amount of roots in the shortest possible time, within 30 to 60 days. And then the most important thing is that it should be resistant to pests and diseases, so that when we use Plectranthus, which is a Coleus species, which grows very fast. The growth is fast, the roots are soft, easy to produce or observe the colonization, and of course is hardly is very much resistant to disease and pest, hardly affected by any disease or pest. So this is one of the species that we have used. We have also used ragi, that is Eleusine coracana, which is a good plant during the summertime to maintain our cultures. Next one, please. So this is the poly house where we maintain the cultures. We have over 35 cultures, pure cultures. All these are pure cultures which contain a single or monospecific culture, we call them. And then when you isolate the roots of this and observe the spores, I try to isolate the spores by taking the roots. You can see the extent of colonization in the roots and the spores which are in the soil. So these spores are fresh spores. They are viable spores. They are young spores, and therefore they are good for preparing fresh cultures or to multiply the cultures. Next one, or to produce the inoculum. Now I'll come to the next one. Yeah, here you can see that these cultures, these are monospecific cultures, a single species. They vary in size, shape, color, ornamentation, subtending hyphae, so many other characteristics which are used for their identification. Next one, sir. Next slide, please. Now you can see the earlier the mycorrhizae were in different classes. So it has been placed under the phylum Glomerate Mycorrhizae. A new phylum has been created. There are about more than 175 mycorrhizal species which have been identified so far. And if you see this classification of Schubler and Walker, 2010, which has 4 orders: glomerales, paraglomerales, then archosporales and diversisporales. There are 11 families and 18 genera. Now this is increasing as more and more number of species have been identified and more new genera discovered. The classification would change. It's not a rigid classification as of now. Next, you can see the 2013 classification of Redeker et al. Next one. Next slide, sir. Yeah, where there are 25. This is Redeker, yeah, right, okay. So again, the classification has changed. Genus Seculospora, this family Seculosporaceae. It has only one species. It had only one species till 2015, and we have added one new species from our department, which we got it from sand dunes habitat from Goa. Next one. Yeah, these are some of the pot cultures. I've not added the recent, the pure cultures that we have developed. We have 36 species of mycorrhizal cultures, monospecific cultures, which we are maintaining in the polyhouse. Next one. And this is Seculospora felinovi, which is the species which we have identified from our lab. This is Rhizoglomus irregularis, a species that produces root colonization within 21 days. And you see the amount of colonization, amount of spores that it produces inside the plant root. And these are the spores in the soil. Extensive amount of spores, indicating that this species could serve a better species for inoculum production. Next one. Now coming to the spore types, basically there are different type of spores based on the spore formation. This is glomus or glomeroid spore, where there's a hyphal attachment here. Directly from the hypha, the spore would form terminally. So there are again variations here based on the type of attachment, whether it is septate or non-septate. Then there is an enterophosphoroid spore of the genus Enterophosphora, where it forms a secule. Here there is a formation of a secule from the hypha, and from the secule it extends and it forms the spore here. Young spore is formed, and whatever the nutrients which are available in the secule are absorbed by the roots, are absorbed by the spore. So the function of the secule is to provide nutrients to the growing spore. So this is a mature spore. This is a equilosporoid type of spore formation, where you can see the spore terminally here, but the secule, sporogenous secule here, which is formed laterally. So this secule develops first, and then the hypha grows and forms the spore. So this secule, it provides nutrients to the growing spore, and finally it forms into a mature spore. So that's the difference between this is formed right about the spore is formed right about the secule. Here the secule is formed laterally, and that's how these are the three ways of spore formation which one can easily identify. Next one. So coming to the different ornamentations, we'll go quickly through this. This is Equilospora species. You can see the color, the shape, the ornamentation, and other features, the subtending hyphae, and the features which are very clear, and you can, based on this, you can easily identify the species. Next one. Next one, sir. Yeah, this is again Equilospora Polonia, Equilospora foveata. This is Equilospora scorbicillata. It's very interesting that one is a older spore, this is a younger spore, indicating that there is a variation in the spore color from in younger spore and the oldest spore. Therefore, it lays credence to what I have said, that we need to produce the trap cultures. So when you have the trap cultures and then you try to identify the species, then it is easier. Then it is easier to know the young stage of how the spore look like, the color, and at old stage. Because if you try to take the spore directly from the soil, and if you get this type of spore, you will name it as one species, and the other type, young spore, you will name it as another species. And that's how there have been problems in mycorrhizal research in the earlier years, that people have been identifying new species one after the other. And now, since there is molecular taxonomy, they are setting the things right, that only those species which are distinct based on molecular taxonomy, those species are retained, where the other species are deleted. Can I see the next slide, sir? Yeah, this is a Glomus glomerulatum. You can see the attachment. There are two attachments here. Here also, Glomus multicolae, there are number of attachments, multiple attachments for a single spore. This is Glomus aggregatum. Large number of spores. Here also Glomus macrocarpum. This is a sporocarpic form. There are also sporocarps form. Not only the single spores, but there are sporocarps fruiting bodies which are formed inside, in the soil. So if you get one or two sporocarp, then you can get number of spores, and you can easily prepare the pure cultures by using the sporocarps. Next one. Coming to Funneliformes. Now, earlier there were 6-7 genera. There was Glomus, Gygaspora, Echilospora, Sclerocystis, Enterophosphora, and Endogon. So, and there was, yeah. So now because of as the classification has, there have been molecular studies done, and they are classified. The genera have increased. Many of the species that was placed on the Glomus, they have been formed into new genera, like Funneliformes constructum. It was earlier Glomus constrictum, Glomus geosporum. So then it was Glomus manihotis, Glomus irregularis. So now the new genera have been erected, like Rhizophagus, Funneliformes. There are 25 genera as I have shown you. Next one, please. There are others like Gygaspora albida. This is again Gygaspora. Now the Gygaspora members, they form this type of structure which is called as the suspensor. It's a bulbous suspensor. It's very clear. This is Gygaspora margarita, which produces a bulbous suspensor. Gygaspora albida, which molecular taxonomy, it is found that they are different. But if you see the structure from close, these bulbous stages, these branched or septate appendages, which clearly prove that this is a different species from Gygaspora margarita. Besides, Gygaspora margarita has got fused lamination. The cell wall has got fused lamination. Beautiful lamination one can see in Gygaspora margarita. Next one. Then coming to next slide. Other species like Ricositra gregaria. It was again earlier Scutellespora gregaria. Now it has become Ricositra gregaria, Ricositra verrucosa, Ricositra corolloidea. Depending upon the ornamentation, this has got corolloid ornamentation. These Spora, they have this type of structures which are called as it's called as the germination shield. Fungi, there are two types of germination. Any part of the spore, they'll produce a germ tube and it will germinate. Other is through this germination shield. So it produces a germination shield which has large number of germ tube initials, and the germ tubes can come out from different places. Next one, I'll show more clear. Next. Yeah, I'll show later on. I'll show another picture of this about the germination shield. This is Sclerocystis, the genus Sclerocystis, which has they are sporocarpic forms. They form spores. Now all these spores which were in a sporocarp. This is a broken sporocarp. The spores are arranged in this fashion on the central plexus, which is made up of the hyphal mass here, and all these spores. Now, based on the ornamentation of these spores, the species have been identified. Next one. Next one. Yeah, here you can see the germination shield, and this is Scutellespora nigra, which is a very dark spore. It's very difficult to identify this species because it looks like a particle, like a mud. This is Scutellespora reticulata, which has got reticulate venation. These ornamentations are very clear, and because of that, some of the species which have clear ornamentation, they are easy to identify, and one can easily identify from them directly from the rhizosphere soil. Next one, sir. These are some of the pictures of the scanning electron micrographs of the EM fungi, how they look under the electron scanning or SEM scanning microscope. This is Cephalospora felinovi, the species that we have identified. And these are some of the Soloidea is again a species that have been identified from our department, from my department. Next one. Yeah, we are talking about the germination shield. This is germination shield. It's very clear here. Now the germination shield is the indirect way of germination of the spores. You can see from the germ tube initial, the germ tube has come out. So like this, there could be 5, 6, 7, 8 germ tubes coming out. So then the colonization would be faster. Next. We also get another phenomenon in mycorrhizal spores. Next one. You get a spore in spore syndrome. Yeah, can I have the next slides? Yeah, you get a spore in spore syndrome. Now this particular is an adaptation to protect the spores. The previous one. So there are old spores. The old spores which remain in the soil, the fungus would sporulate in this old spores, and so that to protect it against the predators. In the soil, there are a number of which feed on these spores. They destroy, they damage the spores, they bruise the spores. So this is a mechanism to protect the spores. Next one. This is the next is the Ecolospora soloidea, the species that has been published in Mycotaxon in 2011, which we have identified. It looks like the radiating sun, and therefore we have named it as Ecolospora soloidea. Next one. Next one. There's another species that is Ecolospora felinovi, which we have identified and it has been published in, I think, in 2015, which is a very interesting species because this species have been identified from the sand dunes. Can you show the next slide? From the sand dunes from the North Goa, and it's a very interesting species. There's only one species identified earlier from Poland. So this is the second species which has, which gives some sort of support to the genus Seculospora. So the next slide. Yeah. Now if you see that the similarities is very much similar to that of the other species, that is Cephalospora baltica. Now this has got important features which are distinct from Cephalospora baltica, is that there are three, the outer wall has three layers. This is the wall number 1, this is wall number 2, and this is wall number 3. In case of, so this is one feature. Second is that the middle wall layer of this S1 of the outer wall, the middle wall layer is very, very fine, whereas in case of it is in Baltica, it is much broader. Then the third, this one, it shows the birefringent properties in polarized light, which the Baltica is not showing. So these are some of the features. And again, next one. Yeah, it also shows this one, the wall layers. There's another inner wall layer. Besides the warts that are shown, you can see the warts, the ornamentation, which are 3 to 4-fold higher than in Baltica. So in Cephalospora felinovi, the warts are higher in number, and also the type of ornamentation, the hypal that is formed, is distinct. Although they look very similar in size and color, but there are differences. Like in felinovi, there are stress branches of different inclination relative to the parent hypha, whereas in case of Baltica, they are generally sinuous. So these very fine observations have made us to identify this is a new species. The molecular sequencing of this has been done. Next. But before that, we tried to see whether there are, or whether this particular species produce orbiscles. Next slide, sir. Next slide. So the orbiscles were very difficult to identify. We had to use different methods, and then we could identify these orbiscles, tree-like structures, which are clear by the staining, and therefore we could confirm. And next slide. that it is you see the features here. You can see this is the Philinovi is very closely related to Baltica. So these are distinctly different from this, and therefore we could identify it as a new species. Next one. So it is not that you cannot one cannot produce species. I mean, it is not a fast process because you need to identify molecular sequencing has to be done. A lot of things are required, and therefore it takes time. This is another novel species which has been unidentified. You can see a number of secules form, and finally the spore is formed. So the secules, as I said, they store the nutrients which provide which is provided to the spore. So this type of structure, this is again a novel species which we are yet to identify. Next one. This is another species of Equilospora species. Next one, sir. Next slide. So Equilospora species, here you can see that it looks like the identified species Equilospora, but you can see that the way these ornamentations are convex shaped, which are uniformly distributed and distinctly arranged over the spore surface. So this is again an unidentified species which we are going to identify shortly. Next one. Now coming to the Ectomycorrhizal fungi, I'll go quickly through this. Yeah, this is Pisolates albus, which is an ectomycorrhizal fungus which is found in the acacia plantations. We have been able to culture it in vitro, and then after 21 days, within 28 days, it covers the entire petri plate. And we could also get pelletization in the liquid culture. Next one. Next slide. So these pellets we were able to grow in the culture media. We were able to grow the pellets also in the culture media, and then we tried for the compatibility, somatic compatibility, and found that there is somatic compatibility in this between both these through the culture that is produced through pelletization and through the culture media. And these are the spores. However, you find that the spores, they don't germinate. You cannot use the spores as an inoculum. So we use the fungal mass or the hyphae as inoculum for inoculating the plant species. Next one. In addition to that, the inoculum production, besides the pot culture inoculum production, we have also produced a in vitro culture successfully cultivated, where we select the propagules, either the spores, or we use the vesicles, disinfect, and by using this transformed DNA technique, we are able to culture by using the specialized culture media. There's a monogenic minimal medium and the modified MSR medium. Next one. On the culture media. So these are the roots of chicory and linum and carrot, which are used for culturing. And when you place the spores in the roots, amidst the roots, they would colonize. Next one. So we can run through these slides. Here you can see how the roots have colonized, have been colonized in this hyphae. They grow in the culture media and they produce the spores. So there is sporulation in vitro. And when we stain this, you can get this picture where the spores also get. These are young spores stained. Next one. So yeah, here you can see how the spores they look. This is the normal spore. These are stained spores. So they produce both intraradical spores or terminal spores. You can see that these are the young juvenile spores that are produced. Next one. And you can see the amount of sporulation in the culture media. This is one petri plate which had 25,000 spores in a single petri plate. So that shows that when we get such a sporulation, then inoculum production is here. You can use this. We can enable to use these cultures. Grow them with the carrier medium. We have formulated carrier medium, the suitable carrier which was used to colonize the different plant species. Next one. Sir. Again, you can see the vesicles we have used as propagules. And the vesicles also, they produce the germ tube, and you can see the glistening young fresh spores which are viable. Next one. Now coming to the application part of this, we have done several studies. I will have taken a few studies. Now this is in case of chili, where we found that there was increase not only in the number of the chilies, but even in the size of the chilies. There was a distinct, significant increase because of mycorrhization. And the different species, mycorrhizal species that were used, we found that a particular species, that is Meridoglomus petuniculatum, was identified as the best species for chili. Next one. So also for the ladyfinger. Next one. Yeah. Also, we found that Equilospora strobiculata was the very efficient mycorrhizal um, species for colonization, where there was increase in the number of pods, in the number of seeds in the pods, the seed weight, all these increased because of mycorrhization. We tried in papaya and found that the different species of mycorrhizal fungi from the pure culture. Next one, sir. And found that Glomus mosae was the best species for papaya, which influenced the mineral nutrition. As a result, the papaya showed better growth and better yield. Endographis paniculata. Next slide. We found again, we inoculated with different species of mycorrhizal fungi, and we found that there was significant growth in almost all the mycorrhizal inoculated plants, but the certain species showed better growth, or when inoculated showed better growth in Endographis paniculata. Whereas in case of control, the growth was much less. So we also studied the amount of endographylite, a diterpenoid, and found that when we used inoculated plants with Gygospora albida, then it increased the concentration of, which is an alkaloid. So that means mycorrhization helps to increase the alkaloid. It is all plant fitness. The more the fit the plant is, the more the plant would be healthy from all ailments or all pests and diseases, and also would produce maximum biomass and maximum yield. Next one. We did experiments in ornamental plant like chrysanthemum, where there were number of flowers per plant increased because of mycorrhization. We have identified this species, mycorrhizal species. The number of days that are required flowering reduced, and it is next slide. Next slide, sir. Tegitis erecta, which is again an ornamental plant. The mycorrhization affected early flowering, then produce more amount of flowers per plant. Next one. In case of Procenta infundibuliformis, where we did experiments where the growth in the mycorrhizal plants was distinctly, was significantly increased because of mycorrhization than in the other species. Next. Then in control. Next one, the mangroves, where we have identified a large number of plant mycorrhizal species for the mangrove afforestation programs. We are directly, we associated with the forest department. And here also we found that we developed techniques to study this mycorrhization in Cereops tegal, which is a mangrove which is found in there are only few plant species in Goa of this particular species, and therefore we tried colonizing this by using mycorrhizal species, different mycorrhizal species in controlled and in unsterilized control, and found that the mycorrhization increased the growth. Not only increased the growth, next one, it also increased the roots. Normally in case of mycorrhizal, in case of plants, the spores, the roots, they grow very slowly. But in this mangrove plant species, it was different. The reason is in mangrove plant species, the growth of the root is very, very poor. So the plant would require roots for better colonization, and therefore in here When we inoculated with mycorrhiza, it produces, it showed better roots, more number of roots, and the plant height also increased. So the better rooting has helped better colonization, and better colonization has resulted in increased plant nutrient uptake and increased plant growth. So this particular the technique as well as the inoculum that we have used, I mean we have transferred it to the forest department so that they can multiply it on a massive scale and bring about afforestation of the mangroves. Next one. This is again the growth studies and growth in biomass. You can see that there's increase in, because of this particular treatment, there was a significant increase compared to the controls in case of and so also in the other species, other inoculated treatments also there was increase, but here the growth increase was much significant. Next one. We also use this mycorrhization, the mycorrhizal fungi in the for afforestation of the waste dumps, the mining waste dumps, the tailing ponds, all along the bunds, the steep slopes. Mining, I've been working on mining for the last several years since I joined the university in 1991, and we have done extensive work initially on screening of plant species for afforestation programs on the mine lands, and then later on using mycorrhizal inoculum for growing for afforestation programs. And we found that mycorrhization helps better growth, better survival. Next one. So we raised nurseries at the mining sites of different plant species that were inoculated with mycorrhiza. So we inoculated mycorrhiza at the nursery stage and then transferred it on the dumps. And then the growth was monitored. And we found that it makes a distinct, significant difference in growth and survival rate on the dumps. Next. So next one, sir. These are some of the where we develop the carrier-based inoculum. So once we have the spores in vitro culture, then we try to develop the carrier. So we mix substrates, produce a carrier. Again, we try to study the property, and we were able to raise this inoculum next. And you can see here that the different development of carrier, the different treatments, substrates used for the carrier, it made a difference as far as the colonization percentage was concerned. So it's just using carrier for the sake of carrier is not enough, but we have to really formulate a good carrier. That's important. Here we use Elucin coracana ragi for raising this inoculum. Next one. This is in case of, next one. This is in case of coleus. The plants these plants were treated with the monogenically produced spore. The spore that we produce in vitro, that inoculum was used for, and you can see the growth in coleus, extensive growth, and the root system was also extensive together with relation. Next. Further, if you next one. Next slide. Yeah, you can see the very extensive colonization is seen, indicating that the inoculum that was produced in vitro has been very effective in colonizing and in supporting plant growth. Next one. You can see orbiscles and vesicles. You can see the root colonization when we try to inoculate the inoculum that we produce in vitro of Glomus intraradicis and Funiliformis mosaic in coleus roots. You can see it has distinctly colonized. Not only there is orbiscular colonization, but there is also vesicular colonization and the spore formation. Next one. Another experiment that we did was in Kaju. The next. Yeah. So we used the monogenically produced carrier for inoculation to study the inoculation and growth in case of Keju, Anacardium occidentale. Now this particular plant we had selected because we wanted to do afforestation programs on the sand dunes, and Keju, Anacardium occidentale, is one of the most important plant in the sand dunes as well as in the mining dumps, on the mining dumps. So we found that mycorrhization has affected good growth in Keju plants. So initial colonization would help the plants to grow better and also help the survivability of these species on the both the dumps as well as on the sand dunes. Next, you can see the colonization. The extent of colonization in Keju is heavily colonized. So when there's heavy colonization would mean that the inoculum is effective. The heavy colonization together with the plant growth indicates that the colonization, that inoculum is effective or efficient colonization. So like this, we have done several studies in different areas and different habitats, and we have identified the plant species. The next one, I think that's the last one. No, that's over. Yeah. So we have been doing work on inoculum production because that is what is more important. And as of now, we are also developing a strategy to produce pot cultures which would produce good amount of inoculum without any problems. Because inoculum in pot cultures, getting good amount of spore density in pot cultures is most important. When you have more amount of spores which are young, which are viable, then the inoculum works efficiently, like what I had shown. Yeah, with this, I would like to take up any questions. Any questions from the audience? I don't know what is the.
Yes, participants, if you are having questions, you can ask. Just you will be given 2 minutes for question. Yeah, Pendkar sir. Yes.
Yes.
Sir, you please unmute, sir. Pendkar sir, please unmute.
Sir.
Yes.
Hello, Rajesh sir.
Yes.
Sir. Hello, sir.
Hello, I can hear you. Yes.
Yes, sir. In our area, banana plantation is a heavy acreage, sir.
Yes.
So,
yes.
any work done regarding specific mycorrhizal preparation on banana?
Yes. Yes. One of my students has produced thesis.
Yes, sir.
On banana, mycorrhization in banana.
Yeah.
So we have identified a species. I don't remember exactly. This was done about 15 years back. She has done extensive work on mycorrhization in banana. And there are several papers, sir.
Yeah. Any local specific work can be done, sir, here with your help?
Very much. Very much. It is important that we identify the local species, the native species.
That is what, sir. So, can you assist, sir, in the development of banana-specific mycorrhizal preparations?
Surely. Yes.
Yeah, sir. Yeah, sir. Thank you. Thank you very much.
Yes.
Thank you, sir.
Yadav sir.
Yes, Murani sir. Yes.
Yes. Sir, Roderick sir.
Yes. Yes.
Yes, sir. Whether I am audible?
Yeah, you are very much audible.
Yes. So it is a very nice and lucid, presentation. It was very good. In fact, I just want to tell you, you have very good cultures. Now, sir, like say year after year, for the maintenance of these cultures, we have some cultures with us, but for year after year, we have to transfer it in the same pot, or we can maintain in another pot, or we can maintain in a soil without plant, like that. How it has to be maintained, like?
Yeah, to maintain the pot cultures in the poly house, we have a special, dedicated poly house for these cultures. Now to maintain this, we maintain them in the soil only. I'm trying to develop another medium to maintain it. That is why I said, not only maintaining, but increasing the density of spores. Now, what we do is that we have problem. We have these limitations in universities and colleges that we don't get enough people to, once the PhD student, they go, they will be in problems. So what I have adopted is different method. I give this work to my MSc students, dissertation students.
Yes.
So they maintain the cultures perfectly, and then they check the cultures every year, to see that whether there is any contamination. If there is contamination by endophytes, we try to isolate, dispose, and prepare fresh cultures.
Yes, yes.
And
Because
that is one. So maintenance is very important. Other
Yes.
thing is we tried the in vitro techniques also. You can maintain it through in vitro technique. And
that is very nice. You have shown the slides and very nice, and the number is very high
yes,
in vitro. That is very good. In fact, it is because
and sir, what we do is we use the nutrient medium. We use Hoagland solution.
yes,
There
yes.
also we use a different method. Like when we use the Hoagland solution for initial, nutrients, Hoagland solution with half the strength of phosphorus
Yes.
initially. Then after 15 days or 20 days, we don't add any phosphorus to the Hoagland solution.
So for that one should have a root culturing, tissue culture. Say like you
in
must
vitro.
have.
In vitro,
Yes,
yeah.
yes.
But
That aspect is necessary. We must have that.
Yes, true. But you can maintain it in your polyhouse or where you are
Yes.
maintaining the pot cultures. So you need to check it every year and then raise fresh cultures if there is any contamination.
It's very nice. In fact, I congratulate Professor Gachande, B.D. Gachande sir, because he has organized such a very nice lecture, because that college, science college, is specialized for pathological research. Okay, so they can develop this branch over there. And we have given one of the project to Dr. Jadhav.
Yes, sir. Yes, sir. Yes.
So he can think, and slowly they can develop such type of research wing there in that college.
Yes, I am trying, sir. So I will require your help and help of Rodri, sir, also.
Yes,
I'll be
yes,
happy
yes,
to help you.
yes,
Surely.
yes. We are there. We can help. There is no problem as such. So it is very good. I congratulate Dr. Professor B.D. Gachande sir and Principal and Jadhav and Professor Marathe for this. Dr. Chauhan, it's very good team of research worker over there. It is very nice. So thank you, Roderick sir, for very good work.
Thank you.
Oh, thank you, sir.
Thank you, Murali, sir. Thank you.
Hello, sir. Hello, sir. Hello,
Yes.
sir.
Yes.
Hello.
Yes, Bhale, sir. Yeah, please.
Yes, sir. First of all, I'm very much congratulations to the Science College Nanded, who has organized such a wonderful lecture based on mycorrhizal fungi. Because Professor Rodriguez is a very renowned personality in the field of mycorrhiza, and personally, I touch with Professor Rodriguez sir. And Rodriguez sir, you have presented very nice presentation related to the mycorrhizal fungi. With your touch, already I am working since last 12 to 13 years, because you teach me regarding for this mycorrhizal fungi, and now with your help, I have very there are about 6 students I am awarded under my guidance under mycorrhizal fungi. Sir, one more query regarding as far as the Maratha region, as Maharashtra region is concerned, because police plant is very important plants for the mycorrhizal colonization, especially from your Shanduns area. But as far as Maratha region is there, there is very black cotton soil is there, and I have tried in my level best for choosing chili plant as well as tagetes plant, jawar plant, as well as maize plant for the mass multiplication of this plant. But as compared to this cotton soil and shandun soils, please prefer or suggest us which plant is very preferable for huge mass colonization or mass multiplication of the indigenous species with these crop plants or restoring plant species. Please, sir.
Yeah, you could use, what about the tomato plant?
Yeah, already we have used, sir.
Tomato plant.
Yeah, already used.
Now you need to identify which are the plants in your locality which produces good. I have shown you the characteristic of these host plants. What are the characteristic features?
Yes, sir.
So based on, I don't know whether tobacco is good.
tobacco is not the indigenous or native plants from
Then
Maharashtra.
you have to use indigenous plant only.
Okay, okay. Because
Yes,
we have tried for all solanacea plants, chili, tomato, and brinjal also, as well as jawar, maize, and rice also. Then also tried for the coleus plant, but as far as this cotton soil is there, some drawbacks to be there, not well mass pollination are found there. So here,
So we have
Dr.
a problem
Bale.
in mass colonization.
yes, sir.
We'll work
Bale,
into
sir.
it.
Yes, sir. Yes, yes.
Bale, sir.
Yes, sir. Yes, sir.
Sir, you can go for some of the plant species, though they are wild, which have a very extensive root system and very short span of lifespan. So such plant, any type you can just select and just go for, try for this. Even
Yeah,
you
yeah.
can you can give one of the problem to one student by choosing these host plants. Okay.
Okay, okay.
But it
that
must
can be
have
done.
a very extensive root system.
Yes, yes.
We found Tagetes erecta is very promising results we have received recently now. Tagetes.
Yes.
What about ragi? Professor Bale,
Rag-
what
yeah,
about
ragi. Ragi is
Ragi
not
is good.
native plant from Marathwada, sir.
But
Bale
even
sir.
ragi
sir.
is
Bale
good.
sir.
Okay, sir.
You can go for worry. Worry is over here cultivated in some of the area.
Okay,
वरी
okay,
आपण
okay.
उपवासाला जे भाताचा वरीचा भात करतो ना.
Oh.
That you can try. That is also having a very good and it is highly native.
Rale म्हणतो आपण.
You can give this present to a student.
Yes, sir.
Professor Bale.
Yes, sir.
As rightly said, you can give this problem to a student. You can work with some 5, 6, or 10 species and identify the best one.
Okay, sir. Okay, sir. Thank you, sir.
Yes, sir. The topic is very interesting and it will be taking much more time in discussions because another guest is waiting for us. So we will contact personally Roderick sir regarding this matter.
I'm sorry that there was a lot of trouble initially.
No problem, sir. But anyhow, you have managed it and delivered a nice talk, sir. Okay, thank you, sir. Thank you, Roderick sir. Now I will request Dr. R.V. Sangvikar madam to introduce the second guest speaker, Professor Sanjay K. Sharma sir.
Good afternoon to all of you. I'm Dr. Rupa Sangvikar, Assistant Professor, Department of Botany. It gives me immense pleasure in introducing Dr. Sanjay K. Sharma, resource person of today's virtual lecture series held in memory of Dr. K.S. Deshpande sir. Dr. Sanjay K. Sharma, Professor, Department of Chemistry, JECRC University, Jaipur. Sir is internationally recognized, a fellow of Royal Society of Chemistry, UK. He is renowned author and editor of many books and journals. He has contributed 21 books from the internationally acclaimed publication houses, Springer, Wiley, Royal Society of Chemistry, Taylor, etc. There is a long list of his published books, and I would like to specify one of his prestigious book series where he is currently editor of Springer, UK, Green Chemistry for Sustainability. He is also life member of many international professional societies. Sir will be speaking on the topic research publications, ethics, and misconducts. Research publications are the direct reflection of the quality of work of the authors, and it is therefore important to agree upon certain standards and know about ethics and misconducts. So definitely we are fortunate to have such an experienced academician, research publisher, contributor with us who will strengthen our knowledge on this topic. Looking forward to hear from you, sir. Thank you.
Namaskar. Thank you very much, Dr. R.V. Sangvikar ji, for this kind and very generous introduction. Am I audible to all of you?
Yes, sir.
Yes, sir. Yes.
So I'm very grateful and thankful to NES Science College Nanded for inviting me for this very prestigious
Dr. K.S. Deshpande Memorial Lecture Series. I'm thankful to the organizer, Professor B.D. Gachande ji, who contacted me and invited me for this lecture series. So it's a very nice opportunity. Do you have a break of 5 minutes, because just you completed one lecture back to back, another lecture we are starting. Is that okay, everyone?
Yeah, okay, sir. We are comfortable. Please continue, sir.
Okay, okay. Thank you very much. Look, when we talk about research and publications, in my opinion, these two things are not different. These two things are interrelated, compensate each other, and strengthen each other. If you are doing a good research, you must, present it well in the form of good research papers. Sometimes, this is my personal experience, sometimes your research is very good, but unfortunately, you publish it in some substandard journal, especially youngsters. If I'm not wrong, the research scholars and early career academicians are also attending this webinar. So in early career or in some academic pressure or in some hurry, sometimes they are not able to select the right journal. And when you are not able to select the right journal and you submit your good research in some substandard journal, it simply has no impact. And the same time, you will not receive good citation, you will not receive good suggestions, criticism, and all the constructive comments. So it is overall, it is a loss for an individual researcher. So research and publication both goes hand in hand, and both of them compensate each other. So let us start today's talk. Just a minute. Let me share where it is. Oh my God. Is it visible to everyone?
Yes, sir.
Yes.
Okay. Let me minimize this bar. Now, hope it is perfectly visible. So, and when we start talking about publications, two more things are very important, and that is ethics and misconducts. Sometimes we intentionally and sometimes unintentionally, we make some small mistakes, some silly mistakes, and those mistakes are, in long run, it becomes very critical. So I will try to discuss with you, interact with you on all these aspects of research publications and ethics and misconducts, right? So it is not moving forward. Okay. So as you all are aware about new education policy of the country, and this new education policy is actually based on three very important components. One is innovation, another one is skill development, and third one is research. And believe me, research is like enter button in your career. If you want to achieve other two goals, means if you want to achieve innovation, if you want to achieve some skill, research is an enter button. And therefore, research publications are of much importance in this regard. There is some problem. So what is research? Research is a careful investigation or inquiry, specifically through search of new facts in any branch of knowledge. Or I think there is some problem in link, sir. Professor Deshpande?
Yes, sir, yes.
Is there some issue in link?
No, sir, there is no issue in link.
So give me 5 minutes. I can, attend through some other laptop. Will you please give me 5 minutes?
Okay, sir, you can take.
Please, please.
Yes, sir. Yes, yes.
Marathi, sir. Marathi, sir.
एक मिनिट त्याच्यामध्ये सर. हा सर.
होस्ट करून टाका ना सरांना, होस्ट करा बरं.
सर त्यांच्या याच्यात प्रेझेंटेशन मध्ये अॅनिमेशन्स आहेत, त्यामुळे ते बहुतेक डिले होत आहेत.
बरोबर. हा. डिस्प्ले.
अच्छा अच्छा बरोबर.
ते अॅनिमेशन्स प्ले व्हायला वेळ लागतो, त्याच्यामुळे तो प्रॉब्लेम
असेल काहीतरी असेल. हा टेक्निकल प्रॉब्लेम असेल. ठीक.
Hello.
Yes, sir. Yes.
Am I audible?
Yes, sir. You are audible, sir.
Yeah.
Yes, sir.
Now it is visible.
Yeah, no, you are sharing your screen. Yes.
Yes.
Yeah.
Yeah. Okay, sir. Okay.
I'm sorry for a bit technical issue. So as I discussed earlier and started with the note that research is exactly, compensate the publications. And because in new education policy, the research is having very important role. And therefore, research publications are of much importance in this regard. We all know that research is nothing but a careful investigation or inquiry, specifically through search for new facts in any branch of knowledge. Recently, we just attended a very insightful lecture of Professor Bernard F. Rodrigues. He's a senior professor of Goa University, and his experience was reflecting in that lecture how seriously he is pursuing the research, and I'm sure all his serious research is definitely reflecting in his publications as well. So the publications is actually mirror of your research. So it is an original contribution, experimentation to the existing stock of knowledge, or it can simply be defined a task of searching from available data to modify a certain result of theory. Irrespective of any domain, it may be from science, from engineering, from medical, from humanities, for arts, for commerce, irrespective of any domain, the type of research is broadly categorized only into two categories. The one is pure or fundamental research, means if you are trying to answer the question why things happen, the research is a pure research. Your research is a fundamental research. So any research which is a try to answer this question why things happen, it is pure research. And any research which is answering the question how things happen, it is called applied research. So my dear friends, especially research scholars and students, if listening me, you please know what type of research you are doing. Is it fundamental research or applied research? Nowadays, the importance of applied research is going high day by day, because applied research is more important, in which two or more than two subject area or two subject area specialists collaborate with each other and do some research. Such researches are now becoming important and popular. So why research publications are important for an individual? Obviously, it is for professional growth, name and fame, intellectual satisfaction. It is very important for an individual. Networking with peers. When you start researching, when you start publishing your research, you start actually networking with your peer and experts of your area. And when you start networking, collaboration starts happening. You start to collaborate. And as I told you that applied research is nowadays becoming popular, the collaborations are very much required for applied research, because nowadays it is not possible to have all the facilities, all the research facilities at the same place or in the same institution. I'm sure your college is very renowned college and having a good accreditation and having good research facilities, but still any institution is not capable to have all advanced technological equipment or high-end facilities. So in any case, collaboration is very much required. Project grants and funding. This is another thing. If you read all the points one by one, you'll find all the points are interrelated. When you start networking, you will have research collaboration. When you have research collaboration, you will have good project grants and funding. And when you start doing good projects, you will get recognition. And after recognition, it is way to your personal fulfillment. And after personal fulfillment, you are really happy. So this is a path. These are the points what one individual want to achieve in his professional life, right? Now the point is, these are points are for an individual. Then what about institution? in which institution you are working, you are serving, what the institution will get from your intellect, how you will be beneficial for the institute in which you are serving. So research publications are also important for an institution, for enriching the profile of faculty and students, for institutional ranking. I'm sure you all know because you are an accredited college. In INIRF ranking or NAC ranking, the weightage of research and publications is very, very crucial, very, very critical. And if your students and faculty are publishing good journals, good papers, good books, then it will be beneficial in the long run for the institution as well. So national and international accreditations, intellectual property. When your faculty and students start publishing good papers, the intellectual property of the institution as whole, it increases manifold. Again, the institution start networking with peers, the institute start research collaborations, the institute get good project grants and funding, the institute is getting recognition through such research publication, and institutional fulfillment. And finally, overall reputation. Overall reputation of an institution. So if we compare what an individual wants from the research publication and what the institution wants from the publication, you will see there are several points which are common. And these several points are actually important for both the parties, means for an individual as well as for the institution. These common interests are 4: research collaboration, networking with peers, recognition, and project grants and funding. So, my dear friends, and it's my humble submission to the authorities of this college, management people also, I think, listening this lecture. I'm thankful to them for sparing time from their busy schedule. I request you all senior people that please encourage your students, please encourage your young faculty to do good research and to publish good research papers, right? So that it would be, in long run, it would be beneficial for the institution as well. If we go to the 7 assessment criteria which the institution have to comply with in order to get a NAC accreditation, these are the 7 criteria: curriculum aspects, teaching, learning and evolution, infrastructure and learning resources, student support and progression, governance, leadership and management, innovation and best practices, and one more is research, consultancy and extension. My dear friends, all 6 points are very important, but research, consultancy and extension is quite important in terms of weightage. If you see the weightage, marks weightage for the universities, for the autonomous institution and for an affiliated institution, you will find the criteria number 3 means research, consultancy and extension is having maximum weightage. Maximum weightage. So we should think seriously about this criteria and how can we enhance our intellect of the institution. And the key aspects of this criteria are promotion of research, resource mobilization for research, research facilities, research publications, consultancy and awards, extension activities and collaborations. This is all about criteria 3. And in that criteria, the one of the key aspects is research publications, which we are going to discuss in length. Right? Now, questions in everybody's mind. I think everyone attending right now, there are many people and there are questions in their mind that what is the difference between good journals and predatory journal. Yeah, it's very common term nowadays. Predatory journals is a point of discussion whenever we meet with the senior people and other academicians, what is peer review? What is indexing? Impact factor versus site score. It is another very critical question. Many of you and even senior professors, senior academicians are also of this opinion that the journals which are having good impact factors are only the good journals. With all due regards to all senior people, I would like to submit that the journals having impact factors are not only good journals. There are a lot of good journals having no impact factor, but having a good site score. So we'll discuss what is the comparison of these two and how it affect your research. What is DOI? What is citation? What is H index? What is I-10 index? How to write a good research paper? What are the scientific misconducts, etc. These are the questions. If anything else is missing, you may please write me in the chat box. So I'll try to answer all the questions. Okay. So when we start something, actually, I am already thankful to Dr. R.V. Sangyakar for a kind introduction. But in brief, I want to share with you my contribution so far. Over 100 research papers, 20 books from international publication houses. Now its number is 21. Recently, a book came from elsewhere. And edited 39 titles as series editor under Green Chemistry book series of a Springer. Many invited lecture and reviewed hundreds of research papers of international research journal. Among all these contributions, the last point, reviewed hundreds of research papers of international research journals. It is closest to my heart, and it is the basic, the skill, whatever I learned from reviewing many papers, reviewing for many journals, and what type of mistakes people are doing, what type of mistakes early career researchers are doing, and why a good paper rejects, what are the shortcomings. All these things I experienced from this only, right? These are few books which I contributed. Green Chemistry for Environmental Sustainability from CRC Talent Francis. Handbook on Biopolymer from Royal Society of Chemistry. Heavy Metals in Water from Royal Society of Chemistry. Handbook on Ultrasound from CRC Press, Talent Francis, USA. Green Corrosion Chemistry and Engineering from Valley. Advances in Water Treatment from Springer. Biosurfactants from Talent Francis. Wastewater Use and Management from Springer. Green Chemistry for Dyes Removal from Wastewater from Valley. Bioremediation from CRC Press. Green Chemistry in Scientific Literature, a bibliometric study from CRC Press. I'm thankful to CRC Press for giving me space in their spotlight section as a spotlight author and editor. These are few things. I am serving as series editor for Springer. I am fellow of Royal Society of Chemistry. I'm thankful to the Royal Society of Chemistry for awarding me fellowship. And this is the latest book recently published in 2020, Green Chemistry and Water Remediation, Research and Applications from Elsevier. And I dedicated this book to all the, Corona frontline workers, our doctors, our, medical staff and administrative staff, and even teachers, all the frontline workers who contributed day and night during last 2 years. I dedicated this book to them. So from all this, one thing is very clear. Why I shared all these photographs and all these details of my publications. One thing is very clear, that there is no way to get experience except through experience. Means experience is a gradual process. You cannot achieve experience overnight. And that's why the skills for writing a good research manuscript is very critical. And when sometimes we think that we know everything and we can do, at our own, and we can submit a paper to any journal. Look, when you start writing.
Sir.
And yes.
Sir, your screen is not visible. Just it has got disappeared. You please once more share screen.
Oh, yeah. I'm just sharing. Is it visible now?
Yes, sir. Yes, correct.
Okay, okay. So many times we think that when we are writing, we think that we are writing wonderfully well. And it is good thing. Be positive means if I'm writing and I'm not liking my own writing, it is not good sign. But what editor want, what journal wants, right? What reviewers want, that is another story. The people sitting the other side of the table, they are much experienced. They are having greater knowledge, and they know how to examine a manuscript, right? So in that case, again, it is stuck. Let me check. Visible?
Yes, yes.
So here we start with, look, I will try my best to answer all the questions in your mind. So be patient. So types of manuscript, there are 3 types of manuscript. One is research paper, focusing on some specific problem with a specific methodology and presents the original findings and conclusion, right? In many cases, in many journals, there is limitation of pages also there. There is limitation of figures there, limitation of number of tables there, even limitation of number of references there in many good journals. But the fact is, research paper is focusing on some specific problem and present your original findings. Second is review article, focusing on some specific topic, reporting the previous work done, and it would be nicely drafted compilation of previously published literature, right? Now this heading, this type of manuscript is having bigger canvas than a research paper, but is smaller than a book. And therefore, the third type of manuscript is book, which is focusing a particular subject with all the freedom and liberty related to page number, figures, tables, references, etc. So my dear students and friends, only these 3 types are possible of the manuscripts. So research paper may be sub-classified as research note and fast communication and something else. But after all, research paper is research paper, review article is review article, and book is the third one. Now, research paper, when we talk about research paper, it is a written and published report describing original research results. It is the very first document. My dear friends, your research paper, as I told earlier, that research paper is the mirror of your research. So research paper is the very first document which is publicly showcasing your research, and in a form whereby peers of the author can repeat the experiments and test the conclusion. When you publish something, means आपने एक पेपर छाप दिया तो वो आपके हाथ से निकल गया. Now everyone or anyone can verify that paper, verify your research. He or she can repeat the experiment on the basis of your research paper. And if it is good, he or she will refer your paper in their future research. But at the same time, if your paper is not good one, if you did not written it well, or you presented some misleading data, or you presented some confusion you have created through your paper, then it will have negative repercussion, negative feedback, negative reaction. It is quite obvious. In a journal or some other source document readily available within the scientific community. So nowadays, because we are living in an age which is an internet age, it is very simple. Every research, everything is just available on a single click. So if you publish something, it is then and there shared by hundreds and thousands of people at a time. So if it is good research, then it is also shared with people. And if it is bad research, that is also shared with the people. When we talk about publication process, it is a team effort. And there are three different stakeholders: journal, authors, and reviewer. And if we see, there will be communication between journal and author. There will be communication between journal and reviewer, but there will no communication between author and reviewer. This is the ideal standard practice as per the international databases like Thomson Reuters, like Scopus, like DOAJ, like any other IEEE, any other indexing agency, publication houses. This is the standard practice. Someone's mic is on, perhaps. I request all of you to please mute. Thank you. Right. So this is how publication process works.
सर अनम्यूट
या प्लीज म्यूट कर लें. आपको कुछ पूछना है तो आप जरूर पूछें, मैं बीच में रोकता हूँ. किसी को कुछ पूछना है?
No, sir. Problem is solved, sir.
Okay. Should I continue?
Yes, sir. Yes.
Okay. Thank you. So when we see the publication process, it is quite obvious. Author submits a manuscript number assigned, editor assigns reviewers, reviewer give comments to editor, and editor rejects or demand revision. Right? This process, I think everyone is familiar with this process, so need not to discuss comprehensively. But one thing I want to highlight here in this process, that the fifth step where editor rejects or demands the revision, this is the halfway. And at this point, either your manuscript rejects or editor writes you back and demands some revision. My dear friends, if it rejects, no worry, story is over for that journal, but you can submit this paper to some other journal. But in case if editor demands the revision, you please be sure that your paper will be published. Your paper will be published. It is the first tip I want to share with you. If editor demands revision, it means they are interested in your publication. They want some more clarification from you. They want some more revision from you, and they want some answers of their query. If you satisfy their query, if you submit a revised manuscript with the answers of their query, your paper will be definitely published, right? But don't get overwhelmed. Sometimes we get overwhelmed. हम सब कुछ एक साथ करना चाहते हैं, and बहुत जल्दी में होते हैं. We are in very hurry, especially the young generation. I come to know many people, many research scholars, since I am dealing with research scholars on the daily basis. So they are in so hurry that they want to publish a paper overnight, and very fast they want to complete their PhD and want to join some job. So I know this young generation is quite capable and very fast, but in area of research and publication, you should be a bit slow. You should not be in hurry. So first of all, break it down, your whole process into step-by-step process. Avoid complex sentence structure and use simple and clear English. Believe me, when you are publishing a research paper, nobody is there to see your ornamental language. Yes, it should be grammatically correct, but not ornamental. I hope you are understanding my words, not ornamental. The English is simple and clear and grammatically correct, but you should write factually. You please don't write the things of here and there. You just write to the point and focus on your research only. And always keep in mind that the paragraph is the essential unit of thought. My dear friends, whenever we start writing some manuscript, we should start thinking paragraph wise. Don't try to write a manuscript overnight. Please, you write it in different installments. Write one paragraph and after 2 days read your paragraph, and I'm sure you will find 5 mistakes in your paragraph, 5 mistakes at least. Then write next paragraph. Means try to think unit wise, try to write unit wise, and write down the dates. Write down with the due dates. You have to be very particular and strict with yourself. You should prioritize your other assignments, right? Assign a due date to each step, that in how many days you will write the introduction section, in how many days you will write the experimental section, in how many days you will write the results and discussion, like that. So assign a due date for each step and make weekly and daily priority lists, right? And the most important, consult with your research guide at every step of writing. It is very important, very important. Nowadays, I'm not sure, I'm very much sure that in your college, it will not be the culture, but in many places, research scholar is doing research on his own, publishing things on his own without consulting the research guide. And whenever some conflict arises, then only he or she, runs to the guide. That, sir, now please help. So consult with your guide at every step, and please be strict with yourself. Means whatever dates you assign to your, different steps of writing, be strict with that. In a simple research papers, these are the main parts. And now onwards, we'll discuss with each part, and in each part, we will discuss what are the ethical misconducts. This will be the way to, discuss the topic at large. So one by one, we'll take a heading and discuss what type of ethical misconduct involved there. So first of all, title. It is quite obvious that developing the title is your opportunity to attract the reader's attention. And my dear friends, remember, readers are the potential authors who will cite your article or and who will contact you for the collaboration. I am repeating. Remember, readers are the potential authors who will cite your articles and or who will be the potential collaborators of yours in future. So title, agar hum dekhen, if you compare our research paper with a movie, then title is the very first opportunity. First of all, the title is only thing which you can, offer to someone. A good title can be defined as the fewest possible words that adequately describe the contents of the paper, and it should be clear. Title should not be too long or too short, and it should not be very specific. My dear friends, it is a human tendency, a human psychology. Whenever you start browsing the net, you start browsing with simple words. You start browsing with ordinary terms. You never start browsing with specific typical terms. So in your title, try to avoid the specific and critical words, because it will limit your search on Google. And obviously, when your paper is not coming in Google search, your citation will be affected badly. You will not have good citation, right? And just a minute, what is this going on again and again? Now it is visible?
Yes, sir. Yes.
Okay. So, and it does not contain any jargon and abbreviation. Now, authors, it is very important that the author should normally be listed in order of importance to the research. In my opinion, the standard practice of the authors is first name should be of research scholar, second name should be of guide, and third name is co-guide if there, and then after no fourth name. This is the standard and ethical practice. But what happens in our country in many cases, that the first author is guide, second author is co-guide, third author is guide's spouse, fourth author is guide's friend, fifth author is friend's nephew, and sixth author is research scholar. Am I right, sir?
Yes, sir. Yes, sir. Yes. Sometimes this happens.
Right? So it is not good. The list of authors should include those and only those who actively contributed to the research, right? Because each author is responsible for the data given in a paper. You cannot escape yourself in any case of misconduct if found there. Suppose on some paper there is some controversy. You cannot escape from the responsibility. आप उस समय ये नहीं कहेंगे कि मेरा तो नाम इस पेपर में ऐसे ही है, मैं तो I have not contributed. So authorship is actually a responsibility, right? The name of the person responsible for all correspondence should be given clearly. And in the list of authors, one author should be assigned as corresponding author. In case of PhD research scholar, the corresponding author should be the research supervisor, because he knows how to communicate, he knows how to correspond with the journal, and he will be the first point who will receive the comments from the journal and discuss, convey the things to the research scholar to revise or rectify the manuscript. And because of his much experience, he will be quite obvious the corresponding author, right? Now, misconduct involved here is gift authorship. जो मैंने आपको बोला पहले कि guide and then guide's friend and friend's nephew and then spouse and what, etc., etc., etc. It's all called gift authorship, and it is a very, very serious ethical misconduct. If you are just putting someone else's name in author's list just to benefit him or her, kindly don't do, because it may be problematic in long run. Now, there is confusion between first author and corresponding author. Because in our UGC guidelines and selection committee guidelines, there is some API calculations is there, and there is some specific weightage to the first author and the specific weightage to the corresponding author. So this debate is going on that who will be the first author and who will be the corresponding author. In my opinion, and the standard practice is, the first author is usually the person who has made the most significant intellectual contribution to the work in terms of research, designing, study, acquiring and analyzing data from experiments, and writing the manuscript. And it is none other than the research scholar. So research scholar must be the first author, and corresponding author is usually the senior author. In case of a PhD student, the guide should be corresponding author, right? And in many cases, he may or may not be first author. In some specific cases, if suppose you are producing some paper from which is outcome of your research project, in that case, first author and corresponding author may be same. So can first author be corresponding author? Usually not, but occasionally yes, right? Otherwise, first author may be corresponding author and vice versa. ये दोनों हो सकता है, and the misconduct involved is conflict of interest. कभी-कभी ऐसा देखने में आता है, which is quite unfortunate, that conflict of interest निकल के आता है. Or that's why in many good journals, they ask you to submit a letter stating that there is no conflict of interest between authors. I'm sure you are relating the thing, because in that letter, all the authors will sign and submit that letter that there is no conflict of interest. This practice is just started since last few years, because journals are also, they are also becoming wise with the experience, right? Second thing, abstract. Abstract is like the trailer of a film. When you see some trailer of a movie, then only you will decide whether to go to see the movie or not. Similarly, your abstract is also a trailer of your research paper. It should stand alone, and it is the advertisement of your research. In many journals, only abstract is you can access. The whole paper is not accessible freely. But if the abstract is interesting, if the abstract is important for a research or for a research scholar, he will definitely go to read the complete paper. But in abstract, the standard practice is it should not exceed 250 words and should be in a single paragraph. And do not use any reference here. My dear friends, I have come to know many research manuscripts in which author do not know from where they should start referring the literature. And they start putting references in abstract only. No reference is required there. Similarly, no abbreviation should be used in abstract. And if the abstract abbreviation is indispensable, in that case, you must give the full form of the term and give abbreviation in bracket first time, and then only use the abbreviation. Now, the misconduct involved is fabrication and falsification. And how it comes? When there is some conference and you have deadline to submit the abstract, and it is today the deadline, and today only you have to submit the abstract if you want to get some certificate of the conference. People fabricate abstract. People falsify the abstract, right? What they do, they see some similar paper, they see some similar abstract, and they fabricate another abstract by doing some words here and there, some manipulation, some falsification, some fabrication, and submit. And unfortunately, friends, there are so many conferences going on in this country where nobody is there to check the authenticity and plagiarism of such abstract. I'm not saying all, but in many cases, nobody cares. They just interested in the number of registration fee only. And that's why this practice is going on. One more thing, when you normally people write abstract at very first day, पहले दिन abstract लिखते हैं, जबकि ऐसा नहीं होना चाहिए. Abstract should be written at last. When you complete the whole manuscript, when you complete the whole draft, then only you should write the abstract, and it should be written after finishing the complete writing of a paper. Then keywords, 5 to 7 keywords followed by abstract. These 5 to 7 keywords should be best to browse on internet. आपका यहाँ आपकी इंटेलिजेंस देखी जाएगी. Select keywords very intelligently. These 5 to 7 keywords are those words which people browse on internet, so that your manuscript come on screen in Google search. Now, after abstract keywords, the first section comes introduction, and introduction is where you give some background information first, state the purpose of study. Here I advise you to don't write extensive literature review. In introduction section, you just write literature review of last 7 to 10 years, not more than that, right? And in that literature, try to take maximum review articles, because every review article has 200-300 references in it. Okay, so try to refer more review articles. And in this section, please clearly mention your aim of study. If you see any research paper, you will see the last paragraph of introduction section will be like it, that in this study, we are going to discuss so and so and so and so. But don't discuss any result here. You just introducing your topic, you just introducing your research, what you are going to present, but don't discuss any result here. And the misconduct involved is plagiarism. Many times when people write introduction section for their paper, they took some similar paper and take introduction from there, and by manipulating few words, they use the paragraphs as it is. It is plagiarism, simply plagiarism, and you please don't do it. Avoid it. Referring is not an issue. You may refer anybody's research, but you are not allowed to copy verbatim from some paper and use in your own paper. It is plagiarism, right? Then the next section is experimental. In many journals, this section is by the name of materials and method. But in this section, you should give exact technical specification of the materials and tools. Describe what you did exactly. आपने कौन सा software use किया, what machine you use, what software you use, what chemicals you use, what technique you use, what sampling size you use. All these experimental, detailings you should provide here in this section. And if you use a well-established method, it may be necessary simply to mention it by name or to give an appropriate reference. अगर आपने किसी का well-established method use किया है, तो you simply name it. For example, if you use Ohm's law, you just write that as per the Ohm's law. Everyone knows what is the Ohm's law, right? So established method should be mentioned simply by mentioning by name or to give an appropriate reference. And the misconduct involves here is falsification. Many times people mention the things which they have not done, which they have not, contributed, not experienced. आपने जो सॉफ्टवेयर यूज नहीं किया, आप उसके बारे में लिख रहे हैं. आपने जो मशीन यूज की उसका स्पेसिफिकेशन जो आप दे रहे हैं उस स्पेसिफिकेशन की मशीन पे आपने काम ही नहीं किया. Got my point? So this is falsification. If you are doing falsification in this section, you are, my dear friends, you are simply misguiding the future generations. People who will going to refer your paper, they will be certainly misguided because you are presenting false things. You are presenting false facts. So don't do it. Then the most important and most critical part of a paper, which is results and discussion. And this is the most critical part where you discuss your own findings and data. Therefore, it should be very authentic. In this section, you will present your observation, you will discuss your analysis, you will discuss your whole research. And that's why it should be original and authentic. और ये होना ही चाहिए. अगर ये section आपका original नहीं है, तो I'm sorry to say आपका research is very much manipulated. This section should be 100% pure, 100% original. And in this section, you please point out the suggestions for the future study. आपको ये बताना चाहिए, you should insist, you should highlight कि आगे क्या suggestions हो सकते हैं in long run or in future. What can you do so that either you will continue this research or you are suggesting the future generation how to proceed. And that is very important, and that is important from the citation point of view. When you suggest something to the youngers, when you suggest something your coming generation, they will certainly refer your paper in their research papers. Your citation will be increased, your popularity will be increased. You will get good references, you will get good reputation, you will get good contacts. Because when you suggest, people will try to contact you. And that's why how your network will enhance. And in this section, the misconduct involved are FFP, means fabrication, falsification, and plagiarism. I have seen many cases, my dear friends, many cases in which people freely they done these three: fabrication, falsification, plagiarism, and they are quite confident that nobody will know. But as I told you earlier, that in the age of internet, anybody and everybody can know anything about your research, right? So please don't do FFP, fabrication, falsification, plagiarism. This section is entirely yours and should be yours. Now, conclusion. Tell people how your work is different and advanced from others. ये आपकी जिम्मेदारी है, it's your responsibility. आपने एक पेपर छापा है, आप एक पेपर पब्लिश करने जा रहे हैं, and you expect that people should read your paper, people should refer your paper, people should follow your paper. So in that case, it's your responsibility to tell how your work is different from other work present so far. Because its uniqueness, it is quite necessary to judge your contribution on certain merits and parameters, right? Provide a clear scientific justification for your work and indicate possible applications and extension. ये बहुत important है. देखिए, जब आप next paper के लिए जाते हैं या आप कभी suppose आपने किसी project के लिए apply किया, किसी funding के लिए apply किया, तो वहां आप conclusion में अगर आपने कुछ ऐसे points लिखे हैं, ऐसी चीजें लिखी हैं जो कि ये justify करें कि आपकी research का ये application है या ये extension possible है, तो believe me, there are a lot of possibilities of granting good grants, good project grants. If you have already mentioned some applications and extension work in your research papers, they will easily grant you money, right? And suggest some future steps, experiments, ideas, and extension of the work. But remember, Do not repeat abstract here. बहुत से लोग क्या करते हैं? I don't know how they do. Either they don't know the difference between conclusion and abstract, or they intentionally use the same text of abstract in the conclusion. Conclusion and abstract are not same. Conclusion involves the results, the gist of the result, as well as some suggestions for extension works. But abstract is having only and only the crux of the research paper. In abstract, you never mention some suggestions, never mention the future ideas. So there is clear-cut difference between abstract and conclusion. So do not repeat abstract here, right? Now come to the acknowledgement. This is very, this section is quite funny. In many cases, we just acknowledge anybody. We acknowledge our friends, we acknowledge our relatives, we acknowledge our family members. But what to acknowledge? You should acknowledge useful helpers. Useful helpers who do not qualify for authorship. जो authorship के लिए तो qualify नहीं करते, लेकिन जिनका help आपके लिए useful था इस research में. मैंने बताया ना, first author is scholar, second is guide, third is co-guide, then no one. So after that, 4, 5, 6, 7, whatever you wanted to list as author, who are not actually eligible for authorship, you can mention, you can acknowledge them. Number one. Number two, technical and financial help. If you received any technical help, if you received any financial help, if you receive fund from some project, you please mention it in acknowledgement section. Material supply, and you should obtain written permission from the individual concerned. Now this line, people many times ask me that, sir, what is the need to get permission from the individual? हम तो उनका नाम लिख रहे हैं, हम तो उनको thanks बोल रहे हैं, तो they should be grateful. And I say no, they should not be grateful. Suppose, for example, कोई पेपर है और उस पेपर में आपने किसी बड़े आदमी को एक्नॉलेज कर दिया. फॉर एग्जाम्पल मैं अगर नाम लूं, मान लीजिए कोई आपका पेपर है, स्वच्छता अभियान से रिलेटेड पेपर है या न्यू एजुकेशन पॉलिसी से रिलेटेड पेपर है, और आपने प्राइम मिनिस्टर को एक्नॉलेज कर दिया. देयर इज नो हार्म इन एक्नॉलेजिंग प्राइम मिनिस्टर, ही इज द हेड ऑफ द कंट्री. बट व्हेन यू एक्नॉलेज सम such people, and suppose on that paper some controversy arise, so in that case जिनका नाम भी acknowledgement में है, वो भी उनसे भी बात की जाएगी कि आप इन साहब को कैसे जानते हैं जिन्होंने पेपर लिखा है. सुनने में बात अजीब लगती है, बट ऐसा भी हुआ है. तो whenever you acknowledge some senior professor, your head of department, your professor, your principal, your any senior researcher, you just write a 2-line mail to them that, sir, I'm going to submit a paper. Since you have guided me a lot in many ways, I want to acknowledge you in my paper, if you kindly permit. इतना सा लिखना है, इसमें कोई ज्यादा कुछ नहीं लिखना है. And I'm sure, 100% I'm sure the person will respond you back that, yes, please go ahead. And then you put their name in acknowledgement, no harm. But unfortunately, people don't follow such practices. Anyway, now last section is references. References, the style of writing references depends upon the guidelines given by the journal or publisher. It varies from person to person, right? Journal to journal, publisher to publisher. But the references should be relevant, latest, and actually concerned with the topic you are discussing. This is important. Reference style कुछ भी हो, लेकिन reference should be relevant. Don't put references just to increase the number of references. Got it? Don't use references just to increase the number of references. एंड माय डियर फ्रेंड्स, आपको एंड अवॉइड एक्सेसिव सेल्फ साइटेशन। अपने पेपर भी आप ज्यादा साइट ना करें, कम करें। आजकल यूजीसी ने भी एक लेटर निकाला है, आई एम श्योर कि आपके कॉलेज में भी पहुंचा होगा, कि excessive self citation will not be considered for the paper in the paper in which you have excessive self cited yourself. Those paper will not be considered for any appointment, any promotion, any other benefits. So avoid excessive self citation. कम करिए, 2-4 साइटेशन अपना भी डालिए, 2-3 रेफरेंस अपने भी डालिए, बट ऐसा नहीं कि रेफरेंस में 50 रेफरेंस में से 45 रेफरेंस आपके खुद के हैं. It is called excessive self citation, right? Third, use variety of references like books, chapters, papers, websites, newspapers. Variety of reference when we use, anybody can understand very easily that you are a serious researcher. You know the variety of references. You have updated knowledge of your field, of your domain. So please use variety of references. Now, my dear friends, in references, what type of misconduct anybody can, imagine? Can you tell me? In references, what type of ethical misconduct anybody can imagine? Nobody is answering? Okay. Plagiarism is there. Now you will say, sir, plagiarism कैसे होगा? Reference तो reference है, वही reference जो है, जो एक ने लिखा है वही तो दूसरा लिखेगा. Reference में क्या plagiarism है? क्या change करोगे आप? You are right. But plagiarism कब होता है? जब आप किसी एक पेपर के 25 रेफरेंस as it is उठाते हैं और as it is use कर देते हैं अपने पेपर में in the same sequence. That is plagiarism. It is not possible that two different researchers will use the same references in the same sequence and put their references in the list in the same sequence. It is not possible, my dear. It is not possible. Many times when people, write introduction section, when they copy introduction from somewhere, at the same time, they copy references from the paper. And those 25, 30, 50, whatever references they copied, they use those references as it is in the same sequence. And this is not possible. This is plagiarism. And no one will tell you that how references can involve plagiarism. But it is the experience which I have. I have seen many manuscripts in which we found plagiarism in references, right? Now, most important in any research paper, whether it is a small paper or long paper, large paper, good study or bad study, but do not claim more than you did. आप उतना ही लिखें जितना आपने किया है, उससे ज्यादा ना लिखें, क्लेम ना करें. Second, do not generalize from one study or a result. सपोज आप नांदेड़ में हैं, आपने नांदेड़ में रिसर्च किया है, नांदेड़ के एटमॉस्फेयर में आपने रिसर्च किया है, वहां की ज्योग्राफिकल कंडीशन अलग है, वहां का एटमॉस्फेयर अलग है, और वहां की स्टडी के बेसिस पे आप एक जनरलाइज स्टेटमेंट लिख दें that this is the finding for India. You cannot claim, you cannot generalize. It is not necessary that the thing which is true in Nanded is also true in Jaipur. So please do not generalize. And few tips. First of all, cover letter. यहाँ हम सबसे ज्यादा गलती करते हैं, सबसे ज्यादा गलती करते हैं, आई एम सॉरी टू से, कि हम जब पेपर सबमिट करते हैं तो हम सीधा लिख देते हैं कि वी आर सबमिटिंग पेपर, और पेपर अटैच कर देते हैं और भेज देते हैं एडिटर को. जबकि आपको एक अच्छा सा कवरिंग लेटर लिखना चाहिए, बिकॉज इट इज योर फर्स्ट चांस टू स्पीक डायरेक्टली टू द एडिटर. It's your first impression. Use it intelligently. आपको एक कवर लेटर लिखना चाहिए, उसमें अच्छे से अपनी अपनी रिसर्च के बारे में एक ब्रीफ पैराग्राफ लिखना चाहिए और उसका इंपॉर्टेंस लिखना चाहिए, और साथ में you should suggest some potential reviewers. It will help the editor to move your manuscript faster. So whenever you submit a manuscript, be prepared to suggest 3 to 6 potential reviewers. And my dear friends, in many good journals, many international journals, in their portal, when you submit the manuscript online, there is one column where you can mention the reviewers to whom your manuscript should not be sent. This is also a provision. वो आपको ये मौका भी देते हैं कि आप उनके नाम भी लिख दीजिए जिनको आप चाहते हैं कि आपकी मैन्युस्क्रिप्ट ना भेजी जाए. Right? It is a very good practice. It is very transparent practice. आपसे जर्नल पूछ रहा है कि आप पोटेंशियल रिव्यूअर्स भी बता दीजिए और आप वो रिव्यूअर्स भी बता दीजिए जिनको आप चाहते हैं कि जिनको हम आपका मैन्युस्क्रिप्ट ना भेजें. How wonderful it is. So please be prepared to suggest good potential reviewers, and the reviewer should represent at least 2 reasons of the word. ये बहुत इंपॉर्टेंट है. हम क्या गलती करते हैं, जब हम रिव्यूअर सजेस्ट करते हैं तो हमारे ही आसपास के लोगों के नाम डाल देते हैं, हमारे ही जानने वालों के नाम डाल देते हैं. आप कोशिश करें कि आपके सब्जेक्ट को जानने वालों के नाम हो, आपको जानने वालों का नाम ना हो. Got my point? Means the people who are doing good research, who are the serious researcher in your domain, in your research area, you please refer them, you please mention their names. And they should not be your supervisor or close friends. कभी भी रिव्यूअर्स में अपने इंस्टीट्यूशन के लोगों का नाम नहीं देना चाहिए, अपने रिसर्च ग्रुप के लोगों का नाम नहीं देना चाहिए और अपने क्लोज कॉन्टेक्ट्स का नाम भी नहीं देना चाहिए. It is not ethical. Right? And submission of paper, सबसे पहले make sure you have adhered to all of the formatting requirements of your mentor and submit your paper. Always keep a copy of your paper on file and in hard copy. ये मैं हर बार सजेस्ट करता हूँ, मेरे हर लेक्चर में मैं बोलता हूँ, मैं अपनी हर क्लास में भी बोलता हूँ कि even we are living in an internet age, we are living in a technical age, लैपटॉप कंप्यूटर सब हैं, लेकिन फिर भी प्रिकॉशनरी अपने रिसर्च पेपर की एक हार्ड कॉपी आप जरूर रखें. बिकॉज गॉड नोज कोई प्रॉब्लम, टेक्निकल प्रॉब्लम अगर आ गई, अभी आपने देखा मेरे प्रेजेंटेशन में टेक्निकल प्रॉब्लम आई, अगर वो ये प्रेजेंटेशन मेरे पास सेव नहीं होता तो मैं दूसरे लैपटॉप में नहीं कनेक्ट कर पाता. राइट? तो that way. So to save your research, to save your data, keep a copy of paper on file and in hard copy. And misconduct involved, जब आप पेपर सबमिट करते हैं तो क्या misconduct वहां होता है? One is redundant publication and second is duplicate manuscript. Redundant publication मतलब waste publication, worthless publication, not required publication, not important publication. ऐसा पेपर जिसमें नया कुछ नहीं है, जो कि important नहीं है, जो साइंटिफिक कम्युनिटी में कुछ एड ऑन नहीं करने वाला है, such paper are called redundant publication. Many times people just publish just to increase the list of publication. So don't do this. And second thing, duplicate manuscripts. In many cases, you duplicate your data. You duplicate your data and you repeat the same data in 2 different papers. Number 1. Number 2, you submit the same paper in 2 different journals subsequently. It is very common bad practice, bad, ethically misconduct कि आपने एक ही पेपर एक ही साथ 2 जर्नल्स में भेज दिया और आपने अपना दिमाग ये लगाया कि किसी एक में तो एक्सेप्ट हो ही जाएगा और अगर एक में एक्सेप्ट हो गया तो दूसरे से हम विड्रॉ कर लेंगे. This is very wrong practice, very wrong practice. You please submit to only one journal, and if they reject your paper, then only you send the same paper to some other journal. I know everybody is in hurry and people are in hurry and they have no patience, but देयर इज नो वे, आपको ये करना ही पड़ेगा। और अगर आप एक पेपर को 2 जर्नल्स में भेज रहे हैं, एंड माय डियर फ्रेंड्स, बिलीव मी, इट इज वेरी मच, यू नो, एनीबडी कैन ट्रैक इट। आपका जब पेपर भेजा जाता है तो सबसे पहले फर्स्ट स्टेज क्या होती है आपको बताता हूं मैं, एज एन एडिटर आई कैन शेयर विद यू, कि जैसे ही कोई मैन्युस्क्रिप्ट हमारे पास आती है तो पहला काम हमारा होता है उसका टाइटल हम पहले सर्च करते हैं, और उसके कीवर्ड सर्च करते हैं, उसका एब्स्ट्रैक्ट डाल के सर्च करते हैं, और अगर उससे रिलेटेड डिफरेंट पेपर्स हमारे सामने आ गए और उसमें से हमको पता लगा कि उसमें से मैटर एज इट इज कॉपी करके डाला गया है, तो पेपर just rejected, straightforward, just rejected, no second thought, right? So if you submit the paper in 2 journals, it is very risky for your own career, your own future papers. आप उस जर्नल में दोबारा कभी पेपर भेजेंगे तो आपका पेपर कंसीडर नहीं होगा, right? तो what is the ultimate learning from this part of this session is focus on क्वालिटी नॉट क्वांटिटी। आपको ये पिक्चर दिखाई दे रही है, एक जार में बड़ी सारी फिशेस हैं और एक जार में 2 खूबसूरत फिशेस हैं। विच वन इज गुड, विच वन इज अपीलिंग, विच वन इज ब्यूटीफुल? ऑब्वियसली जिसमें 2 फिशेस हैं। सो फोकस ऑन क्वालिटी नॉट क्वांटिटी। एंड हमारे जो सुपरवाइजर थे, हमारे जो गुरु थे, वो कहते थे कि अगर if you are producing 2 research paper per year, 2 good research papers per year, you are a good researcher. और वो बात मुझे आज भी याद है। I did my PhD long back in 98, so आज भी मुझे वो चीज याद है और मैं ये चीज हमेशा अपने स्टूडेंट से शेयर करता हूँ, that focus on quality not quantity. Read more, write less. Read more, write less. ज्यादा पढ़ो, ज्यादा लिटरेचर रिव्यू करो, ज्यादा चीजें आप जो है जब पढ़ोगे, लोगों को जितना पढ़ोगे, ज्यादा पढ़ोगे, उतना ज्यादा सीखोगे। आई थिंक हेयर वी कैन टेक ए ब्रेक ऑफ 5-10 minutes. Professor Deshpande?
Yes, sir, you can.
No, I have no need.
Otherwise continue, sir. You can continue.
Otherwise we can continue?
Yes.
Okay.
Continue.
लोग बोर तो नहीं हो रहे?
No sir, no sir. Please continue sir.
अगर टाइम लिमिट हो तो भी आप मुझे बता दीजिएगा कब मुझे खत्म करना है, क्योंकि आप सब सुबह से आप सुन रहे हैं।
Okay.
Okay. So now this again is stuck. ये बड़ा प्रॉब्लम है आज भाई। यहीं से आना पड़ेगा वापस। Okay. So now a very important section, why a manuscript is rejected. मेरे ख्याल है अभी तक की बातें उतनी इम्पोर्टेंट नहीं थी जितनी कुछ चीजें हम यहाँ डिस्कस करेंगे। हम बड़ी मेहनत करके, सो कॉल्ड मेहनत करके, हम बड़ा अच्छा पेपर लिखते हैं कई बार और भेजते हैं, और सडनली हमें लौटती डाक से आता है कि योर मैन्युस्क्रिप्ट इज नॉट suitable for our journal. It is very sophisticated language which I am using. If you get a reply that your manuscript is not suitable for our journal, it is very sophisticated language, very polished language. But what other things involved? Before that, we should know about how to select a good journal. Look, first of all, मैं आपको एक बात यहाँ clear कर दूँ. Whatever I am sharing with you here, I am sharing on the basis of my experience with the top class journals, top class publishers. I am not talking about the journals which are just mushroomed here and there. There are so many journals जिनमें आप आज पेपर भेजिए, कल शाम को आपको लेटर आ जाएगा कि पेपर एक्सेप्टेड, पैसा भेज दीजिए और 2 दिन बाद पेपर छप जाएगा. I am not talking about those journals, right? So please be specific. So the selection of journals depend upon the scope, prestige, circulation, and frequency of its publication. आजकल बड़ा आसान है, बड़ी अच्छी अच्छी टूल्स बन गई हैं। आप अगर एल्सवेयर के होम पेज पे जाएंगे तो वहां आप अपने आप को रजिस्टर कीजिए, इट इज फ्री रजिस्ट्रेशन। आप ऑथर्स सेक्शन में चले जाएंगे तो वहां आपको वो सजेस्ट करेंगे, यू कैन चूज गुड जर्नल्स एज पर योर रिसर्च। दे विल सजेस्ट यू कि आप इस जर्नल में भेजिए, इसमें भेजिए, दिस इज गुड फॉर योर रिसर्च, राइट? सो द जर्नल शुड बी पीयर रिव्यूड. This is the first requirement of a good journal, okay, that should be peer reviewed. Second, the journal should be well abstracted and indexed in international databases. I think you all are aware about different databases like IEEE, Scopus, Web of Science, right? SCI, all these databases are very important. If your journal is indexed in those databases, it means that is a good journal. Having good impact factor or CITES score, this we'll discuss later on. But yes, if a journal is having good impact factor or oblique and good CITES score, that journal is good. एक एक्सरसाइज हुई है अपने देश में पिछले कुछ सालों में, यूजीसी ने भी एक केयर लिस्ट बनाई है। आप यूजीसी के पोर्टल पे जाइएगा, वहां केयर लिस्ट नाम से, वहां भी आपको रजिस्टर करना पड़ेगा। रजिस्टर करेंगे तो केयर लिस्ट मिलेंगी आपको। देयर आर टू टाइप्स ऑफ केयर लिस्ट। द कैटेगरी 1 of the journals which are just indexed with UGC, just listed with UGC, and list 2 is the list of journals which is indexed with SCI and Scopus. So before submitting a paper, you can refer these two lists also. आप ये दो लिस्ट भी देख सकते हैं। इनमें अगर वो जर्नल है जिसमें आप पेपर डालने जा रहे हैं तो you are bit comfortable, you will be bit confident कि हाँ ये जर्नल अच्छा है, right? But be aware about predatory journals. Predatory journals वही जिनकी मैंने अभी जिक्र किया कि the journals mushroomed here and there, जिन journals ने they are just money making machines, they are not having any peer review, they are not having any rigorous editorial practice, they are not having a good international editorial board, and they are just publishing any garbage. You submit the paper, next day you will receive the acceptance, you pay the money, next day your paper will be published online. Those journals are predatory journals, and to know which journal is predatory or not, you just Google the blacklist given by Jeffrey Bell. आप Google कर सकते हैं blacklist नाम से. This list is coined and updated by Jeffrey Bell. And all fake and scam journals that send phishing emails offering open access publication in exchange of payment, all those journals are listed in this list, and this list is updated on regular basis, right? So they have described by the scientific community, and because they are not indexed in standard databases, any research published in them is effectively lost. अगर आपने ऐसे जर्नल में अपना पेपर छापा है तो माय डियर फ्रेंड्स, वो पेपर आपने, वो रिसर्च आपने, यू जस्ट सिंपली वेस्टेड। उसका कोई बेनिफिट आपको होने वाला नहीं है, किसी किस्म का आपको बेनिफिट नहीं होगा, उल्टा आपका नाम खराब हो सकता है। तो प्लीज बी अवेयर एट द टाइम ऑफ सेलेक्टिंग ए जर्नल। सो अगर हम ब्रीफ में अगर देखें, दिस इज द लिंक व्हिच आई हैव गिवन हियर, दिस इज द लिंक ऑफ द बेल्स लिस्ट। एनीबडी कैन write down and copy and take a screenshot. You can access Bell's list by clicking this link. In nutshell, if we compare as a layman that what is the difference, what is genuine and versus predatory journal. So this is the process of a genuine journal, and this is the process of the predatory journal. I think it is self-explanatory and there is no need to explain it much more. You please see that in predatory journal, two very important steps are missing: editorial board and peer review. These two steps are very dubious, very fake, very, doubtful in predatory journals. So this is the basic difference, because when there is no peer review process involved, how can the quality of the paper be judged? On what basis journal accepted your paper without reviewing? It is just wonder, right? So you being a researcher, being a young researcher, because आपके सामने पूरी जिंदगी पड़ी हुई है, पूरा कैरियर है सामने, तो you should be very aware at the time of the selection of a journal, right? Now the point comes that what is peer review? Peer review मैं बार बार बोल रहा हूँ, peer review होता क्या है, and peer reviewed journal क्या होते हैं, right? Peer reviewed और referred journal, ये दो नाम आप बार बार सुनेंगे, दोनों का मतलब एक ही है. The journals in which peer review involves subjecting the author's scholarly work and research to the scrutiny of other experts in the same field to check its validity and evaluate its suitability. Means आपके सब्जेक्ट के एक्सपर्ट्स आपका पेपर पढ़ेंगे, रिव्यू करेंगे, उसकी शॉर्ट कमिंग्स बताएंगे, उसमें सजेशंस बताएंगे, अगर अच्छी चीज होगी तो उसको प्रेज करेंगे, उसको रिकमेंड करेंगे. That is the process called peer review. And peer review is of three types. One is open peer review. Open peer review का मतलब है जिसमें author and reviewer दोनों एक दूसरे को जानते हैं. Got my point? जहां आपने अपने कॉन्टेक्ट्स डाल दिए, जहां आपने अपने मिलने वालों को डाल दिया, या जहां आपकी आइडेंटिटी रिव्यूअर को पता है. It is called open peer review and it is of no use. इसमें manipulations हो सकते हैं, favoritism हो सकता है, आपको benefit of doubts मिल सकते हैं, कई तरह का आपको benefit मिल सकता है. Therefore, in scientific community, open peer review is of no use. Second is single blind peer review. It is something else, bit complicated, in which author don't know who is reviewer, but reviewer knows who is author. Got my point? Author को मालूम नहीं होता है कि किसके पास मैन्युस्क्रिप्ट गई है, लेकिन रिव्यूअर को पता होता है कि मैन्युस्क्रिप्ट किसकी है, राइट? और ये पता दो तीन तरह से चल जाता है, आपके राइटिंग स्टाइल से चल जाता है और आपके रिसर्च कंट्रीब्यूशन से चल जाता है कि आपने जो पेपर आप लिख रहे हैं, जो प्रेजेंट कर रहे हैं, वो आपकी कंटिन्यू रिसर्च फाइंडिंग है, तो वो बहुत जल्दी आइडेंटिफाई हो जाएगा कि यस, इस फील्ड में तो फला फला लोग काम कर रहे हैं, उनमें से राइटिंग स्टाइल किसका है, ये फला आदमी का पेपर है, राइट? सो दिस टाइप ऑफ रिव्यू प्रोसेस इज बेटर देन ओपन peer review. And the third one is double blind peer review. It is the best peer review. All international journals of good quality, of good impact factor, good site score, good reputation, they use double blind peer review. इसमें ना ऑथर को पता होता है रिव्यूअर का, ना रिव्यूअर को पता होता है ऑथर का, और इसमें जो आपके कमेंट्स आएंगे वो कमेंट्स आपके रियली जेन्युइन होंगे, रियली अनबायस्ड होंगे, और आपकी जो पेपर सेलेक्ट होगा वो आपकी अपनी मेरिट पे होगा, आपके अपने वर्क पे होगा, राइट? सो दिस इज हाउ पीर रिव्यू वर्क्स. नाउ मोस्ट पॉपुलर एंड क्वालिटी डाटाबेसेस, अभी मैं बात कर रहा था कि द जर्नल शुड बी इंडेक्स्ड इन द इंटरनेशनल डाटाबेसेस. सो the most popular databases are Science Citation Index, Scopus, Embase, PubMed, Biosys, DOAJ, Ebasco, Chemical Abstract Service, CABI, BioOn abstracts and indexes. These are the quality databases, right? Top 3-4 are very famous, very renowned, very popular, very important, but each and every one is important. अगर आपका जर्नल, जिस जर्नल में आप पेपर भेजने जा रहे हैं, वो जर्नल अगर इन डाटाबेसेस में अगर इंडेक्स है, इसका मतलब that is a good journal, that I can say, right? Now what is citation? अगर अब आपसे कोई पूछेगा कि citation क्या होता है जी, citation word कैसा है? I don't know how many of you are having Google Scholar account, but if you don't have, I suggest you, I request you to please create your Google Scholar account today only after my lecture, because Google Scholar account will be very beneficial for your career. Whoever will refer your research, that data will be automatically recorded in your Google Scholar account, and you can contact them, you can network with them, you can collaborate with them, you can ask them for help, right? Because if people start citing your work, citation means it is a way you tell your readers that certain material in your work came from another source. The title of work, information of author, page number, publisher, date of publication, all this is called citation. But citation analysis is different thing. The Google Scholar account will give you your citation analysis. It involves counting the number of times an article is cited by other works. आपके पेपर को कितनी बार साइट किया गया, कितने लोगों ने रेफर किया, that is called citation analysis. Second thing, what is DOI? Many of you perhaps know it very well, but few students perhaps don't know what is DOI. It is a digital object identifier, a string of numbers, letters, and symbols used to permanently identify an article. अगर मैं लेमैन की लैंग्वेज में बोलूं तो DOI number is like an आधार नंबर ऑफ एन इंडिविजुअल. जैसा हमारा आधार नंबर है, उस टाइप से DOI नंबर आपके पेपर का आधार नंबर है. It is given by crossref.org, और crossref.org आपके पेपर को एक नंबर देगा. आप नीचे मैंने एक एग्जांपल दिया हुआ है, जो रेड कलर वर्ड्स हैं, दिस इज स्ट्रिंग, दिस इज address of DOI, means the organization. Then light blue colored words are called prefix. It gives you information about the journal, journal's name, journal's origin, journal's language, journal's periodicity, journal subject area. All this information from this prefix, and suffix is for your paper. What is the number of papers, what is the page number, what is the volume number, what is the year, all this information is given in suffix. So this DOI itself is a complete reference of a paper. अगर आपको किसी पेपर का रेफरेंस नहीं पता है, सिर्फ आपको DOI पता है, you just click the DOI और उसका पेपर सीधा खुलेगा. And most of the standard journals use DOIs. They assign DOIs to their papers, and in references, they motivate authors to give DOIs of each and every reference, so that people can easily cite those papers. Now the question comes, what is impact factor? Look, few years back, the journal citation report, previously an intellectual property of Thomson Reuters, but since last 2-3 years, Clarivate Analytics took over, and now journal citation report is published by Clarivate Analytics. It has been integrated with the Web of Science and is assessed from the Web of Science core collection. मीन्स जो जर्नल वेब ऑफ साइंस में है वही जर्नल साइटेशन रिपोर्ट में होंगे. ये मेरा सेंटेंस आप याद रखिएगा, ये सेंटेंस आगे काम आएगा. जो जर्नल वेब ऑफ साइंस कोर कलेक्शन में है, मीन्स जो जर्नल SCI इंडेक्स हैं, वही सिर्फ जर्नल साइटेशन रिपोर्ट में इन्वॉल्व है और उन्हीं को इम्पैक्ट फैक्टर दिया जाता है, ये आप याद रखिएगा। Impact factor was coined in 1950 by Eugen Garfield, the founder of the Institute for Scientific Information, and annual impact factor is a ratio between citation and recent citable items published. अब देखिए इसका बड़ा कॉम्प्लिकेटेड कैलकुलेशन होता है, वो कैलकुलेशन समझाने के लिए मैं आपको एक एग्जांपल देता हूं। The impact factor of a journal is calculated by dividing the number of current year citations to the source items published in the journal during previous 2 years. समझिएगा इसको कि किसी पेपर, how many citations you have, how many citation a journal has in a year divided by the publications of last 2 years. Suppose, note that 2017.
मराठी सर म्यूट करा, मराठी सर म्यूट करा.
Should I continue?
Yes, sir. Yes.
Yes, continue.
Okay. Now that 2017 impact factors are reported in 2018, they cannot be calculated until all of the 2017 publications have been processed by the indexing agency. इसको आप 2 बार पढ़िएगा, अपने आप से पढ़िएगा, आपको समझ में आएगी. Now see this example. For example, Nature, आपने सबने Nature का नाम सुना होगा, it is big journal. Nature had an impact factor of 41.577 in 2017. और उसका कैलकुलेशन कैसे हुआ? Impact factor 17 is equal to citations in 2016 plus citation in 2015 divided by publication of 16 and publication of 17. Got my point? कि जिस ईयर का आप इम्पैक्ट फैक्टर निकाल रहे हैं उससे पिछले 2 साल का साइटेशन और उससे पिछले 2 साल का नंबर ऑफ पब्लिकेशन दोनों काउंट होंगे और उससे आपका इम्पैक्ट फैक्टर निकलेगा. Here you can see 74,090 citation they received in 15 and 16 and the publication was 1782. So the impact factor is 41.577. Got it? Now this is graphical representation of the same thing. That impact factor जो कि 3rd ईयर में निकलेगा लेकिन बेस्ड होगा पिछले 2 साल पे. ये बात समझिए, डोंट गेट कंफ्यूज. निकलेगा 3रे साल में लेकिन होगा पिछले 2 साल का, राइट? सो 3रे साल में आपका 100 साइटेशन आया और पिछले 2 साल में पेपर छपे 40 + 60 100 पेपर छपे. तो नंबर ऑफ साइटेशन इन 3rd ईयर डिवाइडेड बाय number of publications in previous 2 years. Got it from this graphic? And here the impact factor is 1, right? This is the methodology how to calculate impact factor, right? Now what is site score? Now a very important thing I'm going to share with you, and I'm sure most of you people are not aware about this fact. The journal evaluation metric is quite fair and acceptable globally. It is a term launched in December 2016 by Elsevier as an alternative to the generally used impact factors. Impact factors के competition में impact factor के alternative के तौर पे site score introduce हुआ 2016 में. Elsewhere ने इंट्रोड्यूस किया, and site score of an academic journal is a measure reflecting the yearly citation to recent articles published in that journal, and it is based on the data of Scopus. This is the first difference. अब आप कंपेयर करते चलिएगा. Impact factor जो था that is based on the data of Web of Science, while site score is based on the data of Scopus. Number 1 difference. Number 2 difference, impact factor is based on the publications of previous 2 years, while site score is based on the publication of previous 3 years. ये सेकंड डिफरेंस है कि यहाँ पिछले 3 साल का डाटा देखा जाएगा। ये मैंने कंपैरिजन का एक टेबल बनाया है आप लोगों के लिए फॉर इजी अंडरस्टैंडिंग। एक कॉलम में पैरामीटर्स हैं, एक में इंपैक्ट फैक्टर है, एक में साइट स्कोर है। इवैल्यूएशन पीरियड इंपैक्ट फैक्टर के लिए 2 साल है, साइट स्कोर के लिए 3 साल है। डाटाबेस इंपैक्ट फैक्टर के लिए जर्नल साइटेशन रिपोर्ट है जो कि वेब ऑफ साइंस पे बेस्ड है। साइट स्कोर के लिए स्कोपस है। एक्सेस आपका इंटरनेशनल इंपैक्ट फैक्टर जर्नल को सिर्फ सब्सक्राइबर एक्सेस कर सकते हैं। You have to subscribe it, you should pay for it, and site score is open access. Evaluated item impact factor में सिर्फ articles and reviews होते हैं, जबकि site score में all types of publications हो सकते हैं। It is the third difference. Third difference का मतलब है कि आपका जो डिनोमिनेटर है वो साइट स्कोर के केस में बढ़ जाएगा, क्योंकि अगर हम एग्जाम्पल लें, नेचर का ही एग्जाम्पल लें, नेचर का इम्पैक्ट फैक्टर था 11.577 बट साइट स्कोर है 14.456। कैन यू इमेजिन? इट्स अ बिग डिफरेंस। और बिग डिफरेंस क्यों है? क्योंकि आपके जो मेथोडोलॉजी ऑफ साइट स्कोर है उसमें डिनोमिनेटर बढ़ गया। साइट स्कोर में डिनोमिनेटर हो गया 7860। मींस पब्लिशेबल आइटम, जितने भी आइटम थे वो रिव्यू थे, पेपर थे, एडिटोरियल्स थे, शॉर्ट कमेंट्री थी, बुक रिव्यूज थे, सब तरह के पब्लिकेशंस काउंट हो गए। और जब सब तरह के पब्लिकेशन काउंट हो गए 3 साल के, तो आपका डिनोमिनेटर बढ़ गया। जब आपका डिनोमिनेटर बढ़ जाएगा तो आपका रिजल्टेंट कम आएगा। दिस इज द ग्राफिक। आई थिंक यू विल बेटर अंडरस्टैंड विद दिस ग्राफिक कि चौथे साल में आपका साइट स्कोर आता है, इंपैक्ट फैक्टर तीसरे साल में आता है पिछले 2 साल के बेसिस पे, साइट स्कोर चौथे साल में आता है पिछले 3 साल के बेसिस पे। तो A is the number of citations received in 2015 divided by B, which is the number of publications during last 3 years, 12, 13 and 14, right? And ये नेचर के साइट स्कोर की कैलकुलेशन देखिए आप। अब आप कंपेयर कर सकते हैं। मींस आई थिंक नाउ ऑनवर्ड्स, टुडे ऑनवर्ड्स, आई थिंक यू विल बी मच कन्विंसिंग एंड यू कैन कन्विंस एनीबडी एल्स। अब देखिए साइट स्कोर में क्या हो रहा है, सेम जर्नल है, सेम नेचर है, 2017 का ही कैलकुलेट कर रहे हैं, लेकिन देखिए साइट स्कोर में क्या हुआ, साइटेशन ऑफ 16, 15, 14 डिवाइडेड बाय पब्लिकेशन ऑफ 16, 15, 14। साइटेशन भी बढ़ गए, बल नीचे का डिनोमिनेटर भी बढ़ गया, और इस तरह से जो रिजल्ट आया वो 14.456 आया है। एंड माय डियर फ्रेंड्स, नोट दैट 2017 साइट स्कोर विल बी रिपोर्टेड इन 2018। रिसेंटली अभी कुछ दिनों पहले 2020 के साइट स्कोर डिक्लेयर हुए हैं, इंपैक्ट फैक्टर डिक्लेयर हुए हैं, और बहुत सारे जर्नल्स स्कोपस की लिस्ट से निकल गए। बहुत सारे लोगों के फोन कॉल्स, बहुत तरह की इंक्वायरी मुझे मिली, मुझे लोगों ने कांटेक्ट किया कि सर हमने जो पेपर छापा था जिस जर्नल में वो तो स्कोपस से ही निकल गया, वो तो अब है ही नहीं, अब हम क्या करें क्या नहीं करें। तो बहुत सारी इस तरह की बड़ी मैस हो गई है। बट यस, देयर मेथोडोलॉजी इज लाइक दैट कि वो 20 का साइट स्कोर 21 में उन्होंने डिक्लेयर किया है, 20 का इंपैक्ट फैक्टर 21 में डिक्लेयर किया है, एंड ऑन दैट बेसिस द रेपुटेशन ऑफ जर्नल इज publicized. Everyone and each one knows about the reputation of any particular journal, whether what citation is, what site score or what impact factor they have. So here I want to say something, that if a journal is not having impact factor, but if it is having site score, good site score, in my opinion, those journals are good journals. अगर आप मैथमेटिक्स के हिसाब से भी देखें, थोड़ी बहुत मैथमेटिक्स भी अगर हम लगाएं, तो आप देखेंगे जिस जर्नल का साइट स्कोर कम होगा, अगर उसका कभी इम्पैक्ट फैक्टर आएगा तो साइट स्कोर से ज्यादा ही आएगा। गॉट माई पॉइंट? वो ज्यादा ही आने वाला है, कम आने वाला नहीं है। तो इफ ए जर्नल इज इंडेक्स इन स्कोपस एंड हैविंग गुड साइट स्कोर, go for it. That is a good journal, right? Now second question is what is H index? ये अभी मैंने आपको थोड़ी देर पहले कहा कि आप लोग अपना गूगल स्कॉलर अकाउंट बनाइए। सर टाइम अगर आप मुझे बताइए, बिकॉज मैं उसी हिसाब से मैं खत्म करूंगा।
Just conclude within 10 minutes, sir.
Within 10 minutes. Okay. That's fine. So H index, you know very well, it is an author level metric, right? आप जब गूगल स्कॉलर अकाउंट बनाएंगे तो आपकी अपनी H इंडेक्स क्रिएट होती चली जाएगी। H इंडेक्स को निकालने का एक तरीका है कि आपके कितने पेपर पब्लिश हुए और आपके हर पेपर को कितना साइटेशन मिला। आपका वो नंबर, वो मैक्सिमम नंबर जो कि कम से कम उतनी ही बार साइट हुआ हो। थोड़ा सा टेक्निकल है, थोड़ा सा समझने में दिमाग लगेगा। The 6 here in this case 6 is H index because 6 most highly cited articles have been cited at least 6 times each. आपके 6 पेपर ऐसे हैं जो 6 के 6 कम से कम 6 बार साइट हुए हैं। That's why your H index is 6, right? And what is I-10 index? I-10 index is, it is another credential. It refers to the number of paper with 10 or more citation. आपके कितने पेपर्स ऐसे हैं जिनके more than 10 citation हैं? दोनों चीजें अलग अलग हैं, दोनों में कंफ्यूज होने की जरूरत नहीं है। H index means वो पेपर जो मिनिमम उतनी ही बार इंडेक्स हुए हों, that is H index. And I-10 index is that number जितने the number of papers with 10 or more citation, आपके कितने पेपर ऐसे हैं जो 10 और more than 10 times साइट हुए हैं, right? ये हम बात कर चुके हैं तो इस पे ज्यादा बात अभी नहीं करेंगे। हाँ, ये जरूर है कि intellectual honesty and research integrity, these two are the basic ethics. और ये ethics आपके ऐसे हैं जो कि रिसर्च में ही नहीं, आपकी डेली लाइफ के भी एथिक्स हैं। Honesty and integrity, these two ethics are of human being. If you are a good human being, you should be honest and your integrity should be undoubted, undoubtful, right? So research में भी ये दोनों ethics माने जाते हैं। And fabrication, falsification, plagiarism, मैंने इस पे बात की। redundant publication पे बात हो गई है। selective reporting, ये एक पॉइंट हम और डिस्कस करते हैं। कई बार हम क्या करते हैं कि जब हम रिपोर्ट करते हैं, जब हम पेपर लिखते हैं, तो जो ऑब्जर्वेशन या जो रिजल्ट हमारे मतलब के हैं, मतलब जो अच्छे हैं, हम उन्हीं को रिपोर्ट करते हैं। जो खराब हैं या जो नेगेटिव रिजल्ट्स हैं उनको हम छोड़ देते हैं। इट इज कॉल्ड सेलेक्टिव रिपोर्टिंग। और सेलेक्टिव रिपोर्टिंग जो है विल बी मिसगाइडिंग, मिसलीडिंग। आप पूरा पिक्चर दिखाई नहीं रहे, आप सेलेक्टिव पिक्चर का एक पार्ट दिखा रहे हैं, तो आने वाली जनरेशन आपके पेपर को पढ़ के मिसगाइड होने वाली है। तो सेलेक्टिव रिपोर्टिंग ना करें। अगर कोई नेगेटिव ऑब्जर्वेशन आया है, अगर कोई नेगेटिव रिजल्ट आया है, उसको भी आप रिपोर्ट करें। नेगेटिव रिजल्ट्स आर आल्सो रिजल्ट्स, ताकि आने वाली जनरेशन उसको ट्राई ना करे, उस पे वो मान के चले कि ये ऐसा नहीं होगा, राइट? तो वो आप ना करें। एंड डेलिब्रेट ओमिशंस, डेलिब्रेट ओमिशन भी एक तरह का सेलेक्टिव रिपोर्टिंग का ही एक पार्ट है। यू कैन से दीज टू आर ब्रदर्स, सेलेक्टिव रिपोर्टिंग एंड डेलिब्रेट ओमिशंस आर ब्रदर्स। अगर आप डेलिब्रेट ओमिशन कर रहे हैं तो भी इट इज अनएथिकल, राइट? इस पे हमारी बात हो चुकी है, इस पे भी बात हो चुकी है। इस पे थोड़ी सी बात कर लेते हैं हम। देखिए, प्लेगरिज्म इज सिंपली एकेडमिक डिसऑनेस्टी, ये हम सभी जानते हैं। and it's simply copyright infringement and is a serious ethical as well as criminal offense. अगर हम लॉ ऑफ द लैंड की बात करें तो प्लेगरिज्म नाउ क्रिमिनल ऑफेंस भी है, एथिकल ऑफेंस तो है ही है, क्रिमिनल ऑफेंस भी है. So just cutting and pasting material from other original work is plagiarism, and it is subject to sanctions like penalties, suspension, expulsion. And if you browse on internet, recently many cases of extreme plagiarism found have been identified in the academia. कई सारे senior professors, even vice chancellor level के लोग plagiarism में listed हुए, penalized हुए, punished हुए. So you may be blacklisted collectively by most of the journals of your field area just on one click. अगर ऐसा एक भी केस पाया गया, again I am repeating, I am talking about standard journals only, I am not talking about mushroom journals. So अगर आप plagiarism के दोषी पाए गए तो आपके फील्ड के, आपके एरिया के जितने भी जर्नल्स हैं, it may be, it is possible that you will be blacklisted collectively, right? So it is that much important. And the ultimate learning from this session, as the wisdom words of Gurudev, that when your intentions are very pure and clear, nature brings support to you. अगर आपकी intention clear हैं, अगर आप pure हैं, अगर आप honest हैं, तो believe me, ईश्वर आपकी मदद करेगा, nature आपकी मदद करेगी, and you will achieve whatever you wish. And these are the last 2-3 slides. How to market your writing. देखिए, जब आपका पेपर छपता है तो ऐसा तो है नहीं कि आपने पेपर लिखा या बुक लिखी और वो आपने अपने अलमीरा में सजाने के लिए लिखी है, राइट? नहीं ना. तो उसको कैसे मार्केट किया जाए? मार्केट वर्ड में भी आपको लगे कि ये थोड़ा सा कॉर्पोरेट से लिया हुआ वर्ड है, बट आई हैव नो शेम इन इट दैट यू शुड मार्केट योर राइटिंग्स. आपको अपनी रिसर्च के बारे में अपने आप आगे होके बताना पड़ेगा. किसी को क्या पता है, किसी को क्या सपना आता है? नो वन ड्रीम अबाउट योर रिसर्च. तो कैसे आप अपनी रिसर्च के बारे में बताएंगे? तो सबसे पहला तरीका है कि आप अपने कॉन्टैक्ट्स को एक मेल लिखें, एक पोलाइट मेल लिखें. माय डियर फ्रेंड, I have recently published a research paper entitled so and so journal in this volume. This is the link given below. I will be obliged if you kindly spare some time to look into it. आप बताइए, ये मेल लिखने में आप किसी को गाली तो नहीं दे रहे हैं, आप किसी का बुरा तो नहीं कर रहे हैं. आपने ये मेल सपोज 50 लोगों को लिखी, बिलीव मी 25 लोग आपकी मेल बिना पढ़े डिलीट कर देंगे, राइट? बच गए 25 में से 10 ऐसे होंगे जो पढ़ लेंगे, जवाब नहीं देंगे. 15 ऐसे होंगे जो पढ़ लेंगे और आपको लिखेंगे कॉन्ग्रेचुलेशंस. और उन 15 में से 5 ऐसे होंगे जरूर जो आपकी रिसर्च में इंटरेस्टेड होंगे, जो आपको लिखेंगे वेल डन, वी विल कोलैबोरेट, और हम कुछ मिलके करते हैं. ऐसे 5 कमेंट्स आपके पास आएंगे. नाउ योर पर्पस सॉल्व्ड. आपने 50 लोगों तक अपनी बात कह दी. जिन्होंने आपकी मेल डिलीट करी है उन्होंने भी अननोइंगली ये देख लिया है कि आपका एक पेपर छपा है. गॉट माय पॉइंट? दिस इज द फर्स्ट थिंग टू डू. सेकंड, ईमेल सिग्नेचर. आजकल ईमेल का जमाना है. आपके सिग्नेचर में, ईमेल के सिग्नेचर में आपके लेटेस्ट पब्लिकेशन का लिंक आप डाल दें. आप दिन में 100 मेल करते हैं, इररेस्पेक्टिव ऑफ योर रिसर्च एरिया. हर आदमी के पास आपका मेल जा रहा है, तो आपका वो सिग्नेचर भी जा रहा है और उसमें जो लिंक है वो भी जा रहा है. तो एनीबडी कैन click the link. If he or she is curious, he will definitely click the link and can see your research. Third, write about your research paper, book on your social media and your blog and website. My dear friends, it is the era of social media. तो अपना जो भी रिसर्च आपने अगर किया है, मेहनत से किया है, अच्छा पेपर पब्लिश हुआ है, so why don't you share the information on social media? एकदम से एक बड़ी क्राउड तक आपका काम पहुंचता है, आपकी रिसर्च पहुंचती है, राइट? And for serious research, LinkedIn is a very wonderful platform where you can share your findings and research. And next, discuss your work whenever you attend any seminar, workshop and conference. जब भी आप कोई लेक्चर अटेंड करते हैं तो अपने काम के बारे में जरूर बात करें, जरूर चर्चा करें, ताकि लोगों को पता चले कि आप एक सीरियस रिसर्चर हैं. एंड गॉड नोज उन लोगों में से कौन आगे बढ़ के आपके साथ आ जाए, कौन आपकी रिसर्च में हेल्पफुल हो जाए. एंड लास्ट बट नॉट लीस्ट, refer your own work in your presentations and discussion and future publications, but be cautious about excess self citation. ज्यादा एक्सेस ना करें, हाँ थोड़ा बहुत अपना रिसर्च अपने फ्यूचर पेपर में जरूर रेफर करें, राइट? सो लर्निंग ऑफ द डे is poor experimentation cannot be masked by brilliant writing. However, poor writing can mask brilliant experimentation. हमेशा याद रखिए फ्रेंड्स कि कई बार आपकी ब्रिलियंट राइटिंग भी आपके बुरे एक्सपेरिमेंट को ढक नहीं सकती है, बट आपकी पुअर राइटिंग आपके ब्रिलियंट वर्क को स्पॉइल कर सकता है. सो ऑलवेज रिमेंबर इट, एंड थैंक यू वेरी मच फॉर लिसनिंग पेशेंटली. I'm thankful to the organizers for giving me this much time, this opportunity to interact, and we can take some quick questions if anybody want to ask. Otherwise, I think I have answered maximum questions.
Yes, sir. Thank you very much, sir.
Thank you.
Yes, sir. Some participants were demanding PowerPoint because it was a very nice presentation. So if you permit, you can share with Dr.
I will definitely share with the organizers.
Yes, sir. Yes. Okay. Sir, thank you very much for your deep insight on the research paper writing, conducts, and ethics. So you have also explained the importance of what you can say, impact factor, citation index, H index, etc., in academic writing. Now the last part is vote of thanks. So I will request Dr. Praveen Paul sir to for vote of thanks. Dr. Praveen Paul sir.
I am reading the comments. I am thankful to all the people, all the participants for mentioning such a lovely comments. Thanks everybody for listening. Thank
Yes,
you.
sir. Yes, sir. So we all are reading your comments, sir.
Yeah.
Yes.
Good.
Yes, very good, sir.
Thank you, Dr. D.M. Dada, sir. Good afternoon to one and all present here.
Praveen sir, mute sir.
I express my heartfelt thanks to Honorable Dr. Venkateji Kabde, sir, President of Nanded Education Society, Honorable Dr. Praveen Patil, sir, Vice President of Nanded Education Society, Honorable Mrs. Shamal Patki, Madam, Secretary of Nanded. Agrawal, sir, Joint Secretary of Nanded Education Society, for accepting our invitation. I am also thankful to Principal Dr. D.U. Gawai, sir, for permitting to organize this lecture series. I extend my gratitude to our both resource persons, Professor Dr. Bernard F. Roderick and Professor Dr. Sanjay K. Sharma, sir, for accepting our invitation as a resource person and delivering very informative lecture in today's lecture series. I am very much thankful to all the delegates, participants, and students of Dr. K. Deshpande, sir, for attending this lecture series. I must mention our deep sense of appreciation to Professor Gasande, sir, for successful arrangement of this lecture series. As we know that no program can be successful with a single person, I extend my thanks to all teaching and non-teaching staff of our department. Lastly, I am thankful to Dr. Mohsina Madam and Mr. Ganesh Ghator for their technical support. Now, with the permission of organizing committee, I declare that the today's K.S. Deshpande Memorial Lecture Series is over. Thank you.
Feedback announcement kara. Feedback.
Feedback.
I request all
Dear participants, feedback.
Thank you very much. And one suggestion from my side.
Yes, sir.
Can I say something?
Yes, sir. Yes.
हालांकि आप फीडबैक का एक सिस्टम आपका है. From such sessions, I request to contributors as well that you please give your detailed feedback, because the organizers organize this wonderful lecture series. It is all for your benefits. So what type of lectures you want to attend, what type of things you want to attend, what are the good things, what are the bad things, what are the technical issues, these things should be mentioned in the feedback. Because as an organizer of such events, we always encourage people to give any silly feedback, even anything, whatever you have in your mind, you just give it to the organizers. You will definitely help them to organize better events in future. So with these words, I thank once again the organizers and contributors. Thank you very much.
Thank you for your suggestion.
Marathi sir, link taka feedback link.
Okay, sir.
So feedback link will be given on Telegram group. So you kindly all fill the link so that you will get the certificate of attendance. Thank you.
टाकली चालते chat box.
नाही सर, चॅट बॉक्स आता बंद होईल. इथंच टाकू आपण.
विचार टाका टेलिग्राम ग्रुप.
टेलिग्राम ग्रुप वरती चॅट बॉक्स मध्ये टाकतो आता 2 मिनिटासाठी.
थँक्यू सर.
चॅट बॉक्स साठी पण टाकली आणि टेलिग्राम मध्ये पण टाकली.
ओके
सर, ते एक अनाउन्स करा तिथं. या लिंक मध्ये एक लिस्ट आहे. त्या लिस्टच्या नंबर नुसार सर्टिफिकेट आहेत. म्हणजे after filling this feedback form, you have given a link of certificate. You just download your certificate from the given link after submission of the feedback.
Okay, sir. So now you end, sir. Meeting host.
Okay, sir. गजानन सर, एंड करू का? हो. Okay. Thank you all.